Fellow sapiens, dim the lights, let your shoulders soften, and take a slow breath with me. We are going to move gently through a long, quiet story. I am your soft-spoken guide. Together we will listen to the small things that keep memory. This is a calm, evidence-first hour for curious minds. Stay with the cadence, sensory image, patient context, careful facts, and an honest accounting of what remains uncertain. If you enjoy slow, factual storytelling, please like or subscribe. It helps make more long, gentle episodes possible. Tell me softly in the comments, how and when do you listen? At night, on a walk, or while doing the dishes? I will leave timestamps in the description so you can find parts easily if you fall asleep somewhere along the way. Now, let's begin. Cold open, toothlight and tide. The slab of limestone comes up at dawn, wrapped in mud and cold. A foreman wipes his hands on his trousers and points, and for a long moment nothing looks remarkable. A weathered surface, a scatter of flint, a shadow where roots once were. Then the shovel lifts a fragment of bone, and the lab technician notices the tooth. It is small, the kind of thing you could pin on a thumbnail. Under a thin film of mineral, it glitters like a bone relic of a sunless sea. The dig notes are careful. Layer 8, a depth measured in centimeters, associated charcoal flecks, and a scatter of broken stone tools. The clerk writes the coordinates. The tooth is bagged, labeled, couriered cold, and wrapped again in sterile paper. At every step, someone says the same thing in a low voice. Handle with care. 2 weeks later, in a lab whose windows face an indifferent sky, the tooth rests under a lamp. The room smells faintly of solvents and warm plastic. A gloved hand turns it. Under the stereo microscope, the enamel shines like old glass. Enamel is the hardest tissue the human body ever makes. It protects a memory. The technician taps a protocol on a tablet, and the tooth will be cleaned, sampled, and if the sediment cooperates, teased into surrendering a set of peptides, tiny fragments of ancient proteins that have survived far longer than DNA might. Imagine for a moment holding a whisper. That is what this tooth offers, a whisper of molecules that once helped build its enamel. Those molecules, amelogenin, enamelin, and others with quieter names, carry sequences, short strings of amino acids that can vary subtly between close relatives. If we can read those strings carefully, they help us place the tooth on the great family tree. They do not tell the whole story, they tell a thread of it. Our central question sits like a pale pebble on the tongue of that tooth. How much can the proteins preserved in enamel change the story we thought we knew about human lineages 1 million or 2 million years ago? Can these stubborn fragmentary molecules shift where we put certain fossils on the tree of life? And importantly, what are the limits of that evidence and how do we test those limits? Pause with that question. It is both simple and large. In the next part, we will walk back to the place where the tooth was found, the dawn light, the soil layers, the first careful notes. We will learn how context makes the molecule meaningful. For now, breathe in the image of the enamel under the lamp, small, glinting, and patient. And imagine the slow work of translating a chemical whisper into a sentence about deep ancestry. In the next part, we'll go to the field notes and the layer that held our tiny witness, the landscape, the sediment, and why context matters before any molecule is trusted. You remember the lamp and the enamel? Now, walk back with me to the place where the soil still smelled of last night's rain, where the tooth first left the earth. The morning on the site is thin and cool. A ridge of light cuts across the trench. Gloves slap gently against trowel handles. Someone murmurs a coordinate into a handheld GPS. Elsewhere, a camera clicks in a regular, steady rhythm. Archaeology is a choreography of small movements. Each one is a promise that what is lifted will be recorded, not merely taken. The tooth was not found in a plastic bag with a label already printed. It was found in a context, layer 8, as the dig book records, a compacted horizon of silt and sand, mottled with gossiping flecks of charcoal. Nearer the edge of the trench, a cache of flaked stone lay in a crescent, napped with deliberate strikes. A few meters away, a cluster of animal bone hinted at meals long finished. Context matters more than anything else in fieldwork. A tooth without context is a beautiful object. A tooth with context is evidence. The layer gives us time. The tools give us behavior. The charcoal suggests fire in the vicinity. The relationship between these items, spatially, stratigraphically, and chemically, is what allows later labs to turn a sequence of amino acids into a sentence about the deep past. Let us slow down and look at the precise gestures that make the context reliable. First, mapping. Before the trowel lifts the tooth, someone photographs it from multiple angles with a scale bar in the frame. A small placard sits next to the find. A total station or a handheld GPS records the three-dimensional coordinates. Those images will later anchor photogrammetry models, three-dimensional reconstructions of the moment of discovery. In a lab, the technician will scroll through those images and say, quietly, "There, that's where it was." Second, stratigraphy. Excavators peel back layers in thin, deliberate sheets. Each layer is described: color, texture, inclusions. A lens of ash is noted. A darker patch may be a pocket of organic matter. The tooth's depth, relative to nearby features, provides a first-order temporal constraint. If it sits beneath a flowstone or a datable tephra, a layer of volcanic ash, that layer may provide a minimum or maximum age. If it sits above a unit with a known paleomagnetic reversal, that too becomes meaningful. For deep-time questions, a million years and more, these broader stratigraphic markers are often the scaffolding we lean on. Third, sampling for dating. The same brush that reveals a tooth will also coax out charcoal flecks for radiocarbon, but radiocarbon only reaches back about 50,000 years. For older horizons, we turn elsewhere. Uranium series on speleothems that cap or underlie archaeological deposits, optically stimulated luminescence, OSL, on sediments that records when grains last saw light, or paleomagnetic signatures locked in clays. On the site, technicians take careful sealed samples from stratigraphic contacts and label them with the same deliberate care as they label the tooth. Later, date ranges will be proposed and refined. A single number seldom holds in deep time, and we will return to why multiple, independent methods are essential. Fourth, microcontext. Small findings can be moved by burrowing animals, by frost heave, by later human activity. The tooth might show signs of being truncated, of root damage, or of abrasion that suggests transport. The excavator notes these details. A photograph with scale, a sketch of the local microtopography. These small notes determine whether the tooth is in situ, still in its original position in the layer, or secondarily deposited into a younger fill. For proteomics, this distinction is critical. If the tooth was reworked, its chemical signatures may be complicated by the chemistry of its new home. Fifth, initial preservation and packaging. The tooth is gently lifted, wrapped in clean paper, and placed into a labeled tube or envelope. The team notes its immediate environment: moist, dry, calcareous, acidic. If the soil is acidic, organic molecules may be less well preserved. If calcareous, mineralization may be stronger. The tube is sealed. Chain of custody forms are signed. Photographs of both sides of the tooth go into the file. The parcel is placed in a cool container for transit. And sixth, human steps and ethics in the field. The site may sit on a living landscape with modern descendants who care for that land. Before invasive sampling or removal, teams consult stakeholders, local communities, cultural custodians, landowners, and heritage authorities. Respectful archaeology treats the land and its stories with attention. Even the choice to remove a small tooth for lab work is not purely technical. It is also a social decision. We will return to this ethical dimension later in the program. Why is this level of field care necessary for a tiny molecule-based study? Because the lab's conclusions rest on probabilities assembled from context. Mass spectrometers and peptide sequences do exquisite work. They do not, however, adjudicate poorly recorded context. A confident proteomic match to a hominin lineage is only as convincing as the stratigraphic and taphonomic story that frames it. Imagine two teeth with identical protein fragments found in different contexts. One sits sealed under a dated flowstone, the other was recovered from a disturbed trench backfill. The first carries a weight of evidence that the second cannot match. The difference lies not in the molecule, but in the chain of custody and the sedimentary tail. In practical terms, when this tooth arrived at the lab, it carried a small paper sleeve with field notes tucked inside. Alongside it came a memory card of photos and a separate envelope with sediment samples taken within a few centimeters. The lab technician unpacked these in a low hum of fluorescence. The notes told the story of the layer, the photos showed the tooth's last moments before extraction, the The sediment provided material for OSL and for geochemical assays. We pause here on the idea of sediments. They are not inert backgrounds. They are active chemical agents that can leach or insert elements into bone. For proteomics, diagenesis, the chemical alteration of biological tissues after burial, can change amino acids, cause cross-linking, and create patterns that labs must recognize and control for. On the field sheet, the sediment's pH, carbonate content, and visible staining are therefore relevant details. Later, in the methods and evidence parts, we will unpack how the lab distinguishes genuine ancient peptides from modern contaminants and from chemical byproducts of long burial. For now, hold the scene. Dawn in a trench, the tooth cradled in clean paper, field notes in a careful hand, and a set of sealed samples whispering promises of dates. The work in the field is slow and exacting because the questions in the lab are easily misled. A single mislabeled sample can send a confident conclusion astray. In the next part, we will open the microscope of the lab and look closely at what a tooth keeps inside, the structure of enamel and dentine, the proteins that build them, and why enamel is such an extraordinary archive. Dim the lights a little more and bring the image of that tooth back into focus. Hold it in your mind as a small, patient object, not merely a fossil, but a layered archive. We will look inside it now, slowly and with care. At first glance, a tooth looks simple, white, curved, familiar. Under a hand lens or a microscope, it becomes intricate. Tooth tissue is organized into a few distinct parts, each with a different story to tell. The crown, the part that chews, is sheathed in enamel. Enamel is astonishing. It is the hardest biological substance humans ever make. It is mostly mineral, crystals of hydroxyapatite arranged into tightly packed rods called prisms. Between those crystals and within their tiny boundaries once lived proteins. Enamel formation is an act of construction carried out by specialized cells called ameloblasts. These cells secrete a matrix of proteins that guide mineral crystals into ordered arrays. As the enamel matures, much of the organic matrix is removed and the mineral load increases. The result is a near-immortal shell that protects inner tissues. Because enamel is so mineral-rich, and because it becomes essentially a cellular after formation, it is unusually good at preserving molecules that become locked in the mineral matrix. Think of it as a museum that closed its doors early and left a few artifacts behind. The proteins that remain, fragments of the original enamel matrix, can be embedded tightly within crystal boundaries, sheltered from the microbial storms and chemical weathering that degrade many other tissues. Beneath the enamel lies dentine. Dentine is softer and more organic than enamel. It contains a collagenous matrix threaded with microscopic tubules. Because dentine retains an organic scaffold, it sometimes preserves a different suite of molecules. Collagen peptides, perhaps traces of ancient blood proteins, and chemical signatures that record diet and stress. Dentine can be more chemically reactive than enamel in some burial environments. In other circumstances, however, the collagen within dentine is the last substantive molecular trace available when enamel proteins are sparse. At the root, cementum seals the tooth to the jaw. Cementum can preserve incremental layers that tell a seasonal tale of growth. Dental calculus, hardened tartar on tooth surfaces, becomes its own microarchive. Mineralized plaque-trapping microfossils, starch granules, environmental debris, and sometimes DNA from oral microbes or the host. Each tissue is a different kind of recorder. Now, let us listen to what those tissues can tell us and how. Enamel proteins, names you will often hear in the lab, include amelogenin, ameloblastin, and enamelin. These are structural and regulatory proteins essential during tooth formation. They are not abundant in a mature, fully mineralized enamel slab, but the fragments that persist are informative. Because these proteins have sequences encoded in the genome, their amino acid compositions and sequence variants can reflect evolutionary differences between species. Proteomics, the study of proteins, seeks to extract those peptides, read their sequences, and compare them across specimens to infer relationships. Dentine contributes complementary information. Collagen peptides from dentine have been a mainstay in paleoproteomics outside the human lineage. Collagen is sturdy and its sequence evolves slowly, making it useful for deep comparisons. In hominin studies, enamel proteins are often more informative because they are more likely to survive in many burial chemistries and because enamel is harder to replace or disturb. Two features of tooth tissues make them especially valuable for deep time studies. First, incremental growth. Teeth grow in layers. As enamel and dentine form, they lay down incremental marks, lines that record developmental tempo. In human teeth, these can include parencymata on the enamel surface and internal daily or weekly growth lines. These lines are like tree rings. They provide life history data, the age at which a stress occurred, the pace of childhood growth, perhaps even the timing of weaning or illness. For ancient teeth, these lines help anchor biological events to the individual life of the animal or person. Second, chemical entrapment. Mineral crystals can occlude peptides, effectively entrapping them for very long periods. Unlike many soft tissues that collapse and decay, the mineral lattice in enamel acts as a microcage. Molecules within these cages are accessible to modern mass spectrometers, but only if the lab takes pains to extract them without contamination. That brings us to a constant whisper at the edge of every promising sentence in paleoproteomics: contamination. Modern proteins are everywhere. Human handling, lab reagents, environmental microbes, all can leave modern peptides that confuse the signal. The labs that do this work place great emphasis on cleanroom protocols: gloves, filtered air, blank extractions, and rigorous procedural controls. They monitor for markers of modern contamination, compare sequences to expected evolutionary patterns, and look for damage profiles consistent with long-term burial rather than fresh molecules. The most convincing claims couple careful field context, strict chain of custody, and lab practices that make modern intrusion unlikely. A second technical caveat is diagenesis, the post-burial chemistry that alters tissues. Amino acids can racemize, change chirality, cross-link, or be chemically modified. These changes can make peptides harder to read, or can produce fragments mistaken for ancient sequences. Analysts therefore look for patterns of damage that match known diagenetic signatures. When possible, multiple labs replicate the work to ensure the pattern holds. We should also speak quietly about timescales. DNA degrades relatively rapidly on geological timescales. In many bones older than several 100,000 years, DNA is fragmentary or absent. Proteins, particularly those bound within mineral matrices, can persist beyond the reach of DNA under favorable conditions. That is why proteomics can reach back to epochs where genetic material is no longer retrievable. But persistence is not uniform. The survival of peptides depends on burial chemistry, temperature history, and the original tissue's microstructure. In different contexts, enamel may outlast ancient DNA by orders of magnitude. In others, it may not. What does a working lab do with a small tooth fragment? They take a conservative cut. A tiny chip of enamel is removed, cleaned, and subjected to chemical extractions that release trapped peptides. Those peptides are then analyzed by mass spectrometry, which reports the mass and fragments of peptides. From those data, sequences are inferred and compared to reference libraries. These comparisons are probabilistic. They provide statistical weights for different evolutionary placements. A confident placement is one where the sequence matches expected patterns for a lineage and where alternative explanations, contamination, diagenesis, mislabeling, are unlikely. Finally, remember the human and ethical dimension. These are often human remains. They come from landscapes with living people who may see the remains as ancestors or as part of sacred geographies. The decision to sample a tooth for destructive analysis is never trivial. It requires consent, dialogue, and transparent stewardship. We will return to the ethics of destructive sampling in a dedicated part, but for now, hold this as a quiet rule. Scientific curiosity must be balanced with respect. As we close this part, imagine the enamel again, a crystalline lattice with tiny, precious fragments of a life. These fragments are small, but they can carry echoes of childhood stressors, of diet, of genetic affinity. In the next part, we will turn from what the tooth holds to the question that drives much of this episode: why proteins sometimes outlive DNA, and what that means for rewriting the deep past. Dim the lights further, and imagine two clocks sitting side by side in a quiet room. One is delicate, its hands made of thin glass. The other is armored in metal and set inside stone. Both tell time, but one will survive a fall better than the other. In the archive of the deep past, DNA is often the delicate clock. Certain proteins, especially those trapped in mineral, are the armored one. You hold that image because it helps frame a central technical reality. Different biological molecules decay by different pathways, and those pathways are shaped by environment. The result is simple to describe and subtle to apply. Let us walk through the mechanism slowly to understand why proteomics can reach where ancient DNA sometimes cannot, and why that reach is both powerful and limited. Start with DNA. It is a long polymer built from nucleotides joined by phosphodiester bonds. Those bonds break. Hydrolysis, the chemical reaction with water, cleaves the backbone, producing shorter and shorter fragments over time. Bases themselves can be chemically altered. Cytosine deaminates into uracil, adenine oxidizes, and crosslinks can form between strands. Microbial attack chews away at what remains. Temperature accelerates all of these processes. Higher temperatures mean faster chemical reactions. Water, pH, and oxygen availability matter too. In warm, wet soils, DNA degrades quickly. In cold, dry, anoxic environments, fragments can survive much longer. Crucially, when DNA fragments become too small and modified, reconstructing longer genetic sequences becomes impossible. The short reads that remain may be noisy, contaminated by modern DNA, or indistinguishable from environmental sequences. For many fossils older than several 100,000 years, and in many climates, DNA simply does not survive in a form useful for reconstructing genomes or confident phylogenies. Proteins, by contrast, are polymers of amino acids joined by peptide bonds. They are chemically different, and in some burial contexts they persist longer. Two main reasons account for this comparative persistence. First, structural entrapment. In enamel, the hardest, most mineralized tissue in vertebrates, proteins can become occluded within the crystal lattice as the tissue mineralizes. Once trapped, those peptide fragments are physically sheltered from the microbial enzymes and the free water that would otherwise catalyze rapid hydrolysis. The mineral matrix acts as a microenvironment where chemical exchange is slowed. This is comparable to sealing a delicate object in resin. The surrounding medium stabilizes it. Second, intrinsic molecular stability and cross-linking. Some proteins form cross-links that increase their resistance to chemical attack. Over burial time, proteins undergo predictable chemical modifications, deamidation, the conversion of asparagine or glutamine to their acidic forms, oxidation, racemization, the conversion of L-amino acids to D forms, and other diagenetic transformations. Some of these changes make peptide sequencing harder, but they also leave a damage profile that signals antiquity. In practice, labs look for those damage signatures as evidence that the peptides are genuinely ancient rather than modern contaminants. But note that proteins last longer is not a blanket rule. Survival depends on the same external variables that govern DNA decay: temperature history, pH, water activity, and microbial ecology. A protein in a hot, acidic bog may vanish faster than DNA in a cold permafrost. What distinguishes enamel-associated proteins is their physical shelter and the tissue's inherent resistance to breakdown. That combination expands the temporal reach of molecular analysis into windows where DNA is sparse or gone. This difference in survival translates into complementary strengths for proteomics and ancient DNA. DNA, when preserved, offers genomic breadth, whole genomes, patterns of ancestry, and fine-scale population histories. Proteins offer depth in other ways. They may survive when DNA does not. They can come from tissues like enamel that are common in the fossil record. And their sequences, even when fragmentary, can contain phylogenetically informative variants that help place fossils within broad evolutionary radiations. Yet, proteomics has limits. The amount of phylogenetic information in preserved proteins is constrained by two factors. One, only certain proteins persist reliably: matrix proteins in enamel, collagen in bone, and dentine. That reduces the set of genes we can interrogate. Two, proteins evolve under functional constraints. Many positions are conserved, and sequence differences accumulate more slowly than in neutral regions of DNA. That means proteomic comparisons often resolve deeper splits cleanly, but may struggle to discriminate among very close relatives. In practice, researchers use protein data to test broad placements and to triangulate with morphological and, when available, genetic data. Another important distinction lies in error profiles. Ancient DNA analysis has well-developed statistical frameworks for handling short, detect damaged fragments and for identifying modern contamination. Paleoproteomics borrows and adapts many of these tools, but faces unique challenges. For example, chemical modifications may mask a true amino acid residue, creating ambiguity in sequence reads. Racemization and deamidation alter mass signatures that mass spectrometers detect. Analysts must model these changes to infer the original sequence. Sophisticated software and manual curation help, but uncertainty remains, and different labs may interpret marginal peptides differently. A related challenge is contamination, but here the shape of the problem differs. Modern proteins are everywhere, from human handlers, lab reagents, and even airborne skin flakes. Because proteomic workflows often target short peptides, a contaminant peptide can sometimes match ancient sequences closely enough to mislead analyses. To reduce this risk, labs apply strict cleanroom protocols, run extraction blanks, and look for damage patterns consistent with long burial. They also prefer proteins that are unlikely to be modern lab contaminants and cross-check identifications against negative controls. Beyond technicalities, there is an epistemic point worth holding gently. Both DNA and protein data are probabilistic. When a peptide sequence supports a particular phylogenetic placement, it raises the probability of that placement, not a certainty. Best practice is therefore integration. Morphological traits, stratigraphic context, isotopic signals, and molecular data together build a robust inference. A single preserved protein that points one way is interesting. Concordant signals across independent data lines are stronger. Finally, think about environments as selectors. Permafrost and caves are friendly to both DNA and proteins because they are cold and stable. Warm, wet tropical settings are less friendly to DNA, which is one reason why proteomics has been transformative for specimens from parts of Asia, where DNA rarely survives. Enamel and dentine are common even in fragmentary remains. They are often the only tissues left to study. The capacity to extract and read their proteins opens doors to questions about deep human lineages, questions that would otherwise remain locked behind time's chemical erasure. We close this part with a careful, unhurried reflection. Proteins are neither superior nor inferior to DNA. They are different tools shaped by chemistry and context. Each molecule tells a part of the story. Together, they let us piece a more honest picture of our past, provided we respect their limits and hold our conclusions lightly. In the next part, we will move from these conceptual ideas to the practical laboratory steps, how a tiny chip of enamel is handled, how contaminants are excluded, and how mass spectrometers read those faint peptide signals in a way that scientists can trust. Dim the light a fraction more, and imagine a small white room with filtered air. The hum is low and even. A gloved hand opens a sterile tray. On it sits a tiny tooth, labeled, photographed, and wrapped with the same respect it received in the field. This room is where molecules are asked their quiet questions. Sampling begins with an ethical decision. A tooth is a human, or hominin, remnant. Before any destructive step, researchers discuss necessity, minimal sampling strategies, and consultation with stakeholders. The aim is to remove the smallest possible volume that still yields useful peptides. A micro drill with a fine burr rather than a saw, a chip no larger than a fingernail rather than a whole crown. These choices are deliberate and often documented in permits and communication records. Once the permission and documentation are settled, the technical choreography begins. Everything in the room is about exclusion, excluding modern proteins, excluding airborne dust, excluding human skin flakes, excluding any modern enzymatic activity that would mask antiquity. Surface cleaning is the first physical gesture. The lab gently removes any adhering sediment and then abrades a thin outer layer of enamel to dig beneath possible modern contamination. Some teams use UV irradiation to degrade surface proteins. Others apply controlled mechanical abrasion or washes with solvent to clear surface residues. The goal is to access the more protected interior of the enamel, where authentic peptides are likeliest to survive. Next comes microsampling. Under a stereo microscope, a tiny chip or powder is collected from the inner enamel surface using a sterile micro drill. The powder is transferred into a sterile tube. At every step, extraction blanks, tubes that undergo the exact same handling but contain no sample, are prepared in parallel. These blanks act as sentinels for contamination. If peptides show up in blanks, they flag laboratory-derived interference. Extraction follows. Because enamel is highly mineralized, the protein fragments are often bound within the hydroxyapatite crystal lattice. To release them, analysts use mild demineralizing agents that dissolve a small amount of mineral without destroying fragile peptides. Common approaches include etiquylation or dilute acid treatments that free peptide fragments from crystal interstices. Some protocols prefer enzymatic digest after demineralization to generate peptides of predictable length. Others rely on direct extraction of endogenous peptide fragments. The technique is chosen with the sample's preservation in mind. After extraction, the sample undergoes cleanup steps to concentrate peptides and remove salts and mineral debris. Techniques such as solid phase extraction concentrate peptides into a small volume suitable for the mass spectrometer. Throughout these steps, extraction blanks and process controls travel alongside the sample to the mass spectrometer. Mass spectrometry is the machine's whisper, a conversation between ions and detectors. Liquid chromatography separates peptides by size and chemical properties. Then a high-resolution mass spectrometer, often an Orbitrap or similar, measures the mass-to-charge ratio of peptides and fragments them in controlled ways to read short amino acid sequences. Tandem mass spectrometry, MS, produces fragment spectra that analysts match to theoretical spectra derived from reference protein databases. This matching step is statistical and conservative. Spectral search engines assign scores to peptide spectrum matches and compute false discovery rates. Researchers often apply stringent thresholds to call a peptide identified. Importantly, in paleoproteomics, these searches explicitly model diagenetic modifications, deamidation, oxidation, and others, which change peptide masses. Search parameters include allowances for those modifications so that ancient chemically altered peptides are not missed or misassigned. Authentication is not solely a computational trick. It is an accumulation of signals. Teams look for damage patterns consistent with antiquity. For example, elevated levels of deamidation on asparagine and glutamine residues, which accrue over burial time. Sequence positions that differ from modern contaminants in evolutionarily sensible ways. A peptide variant that matches an ancestral node rather than modern lab workers' proteins increases confidence. Reproducibility across independent extracts from the same tooth or across neighboring teeth from the same context. Clean extraction blanks with no overlapping peptides. Phylogenetic coherence. When multiple peptides pull the specimen toward the same placement in a tree, that concordance strengthens the inference. Analysts are careful with low-coverage signals. A single peptide, particularly one shared across taxa, is fragile evidence. Multiple taxa-informative peptides with independent damage profiles are what produce confident statements. Reference databases are central. Once peptides are reported, they are compared against curated protein sets from living species and from sequenced genomes where available. For extinct or poorly represented lineages, researchers infer ancestral sequences based on closely related taxa, but they also remain cautious. Reference bias can mislead if a lineage had unexpected sequence changes. This is why proteomics is most powerful when integrated with morphological and stratigraphic data. Cross-laboratory replication is a gold standard. Independent teams repeating the extraction, using different equipment, or analyzing a separate portion of the specimen help rule out lab-specific artifacts. When independent labs converge on the same peptide identifications and similar damage patterns, confidence rises. Statistical models further quantify certainty. Bayesian approaches can weigh peptide evidence, account for possible contamination, and incorporate phylogenetic priors. These models do not produce certainties. Rather, they produce probabilities. A proteomic result increases or decreases the probability that a specimen belongs to a particular clade. It does not alone prove it. Ethical note, brief and important. Destructive sampling is ethically charged. Many institutions now require explicit statements of minimal destructive impact, plans for storing remnants, and, where relevant, agreements with descendant communities or national heritage authorities. Sampling strategies emphasize minimalism and data sharing. When a tooth is sampled, the remaining fragments are curated and documented to allow future reanalysis or alternative tests that might be less invasive. A final procedural note concerns data stewardship. Raw spectral files and processed identifications are typically archived so that the wider community can reevaluate claims. Reanalysis by independent groups is not only allowed still but encouraged. Science is iterative. Proteomic claims that alter phylogenetic placements gain credibility when they survive the scrutiny of reanalysis. As we step back from the technical humming of the lab, allow the image of the microdrill, the sterile tube, and the mass spectrometer to linger. The next part will step away from peptides and into time, how we date the teeth and anchor molecular signals to geological and archaeological chronologies, uranium series, luminescence, stratigraphic cross-checks, and the careful arithmetic of age ranges. Dim your gaze and imagine a handful of numbered tubes laid out like a row of quiet sentinels. Each tube holds a tiny portion of earth, a sliver of flowstone, or a speck of charcoal, and each of them will be asked the same patient question: how old is this place? Dating is the arithmetic that turns a molecule's whisper into a sentence placed in time. Without it, proteomic signals hang in the air like notes without a score. We will walk through the main dating tools used for deep timescales relevant to teeth and caves, and then talk about how scientists combine them into a careful age model. Keep in mind, every method has strengths, limits, and assumptions. The art of chronological work is to stack independent methods so their constraints cross-validate one another. Radiocarbon is familiar and powerful for the recent past, but it reaches only to about 50,000 years under ideal conditions. For the million-year windows we care about, different tools take the lead. Uranium series, youth dating, uranium series methods date materials in which uranium is incorporated and thorium is excluded at formation. Classic examples are speleothems, flowstones, stalagmites, in caves, and sometimes the mineral crusts that cement sediments. Uranium decays to thorium in a chain with a known half-life. Measuring the ratio of uranium to thorium isotopes yields an age since mineral formation. On archaeological sites, flowstones that cap an archaeological layer provide a minimum age for what lies beneath. A flowstone that underlies a deposit gives a maximum age. In cave systems, UT on flowstones is often one of the most precise anchors for middle to early Pleistocene sequences, provided the flowstone has remained a closed system and has not later altered its uranium content. Lab checks for open system behavior and careful sampling of pristine layers are therefore essential. Electron spin resonance, ESR, on tooth enamel ESR measures trapped electronic charges that accumulate in mineral lattices over time as a consequence of natural background radiation. In tooth enamel, ESR directly estimates the time since the enamel was buried and shielded from light. Because uranium may move into and out of a tooth after burial, ESR is often paired with U-series measurements on the same tooth or nearby materials to model uranium uptake history. This combined ESR/U-series approach provides a direct age estimate on the tooth itself, which is powerful because it ties the dated signal to the specimen that will be studied for proteins. Optically stimulated luminescence, OSL dating applies to the sediments that surround finds. It measures the time since mineral grains, usually quartz or feldspar, were last exposed to sunlight. When sediments are buried, their trapped charge resets. Subsequent burial accumulates a luminescent signal that can be read in the lab. OSL is especially useful for contexts where you have well-preserved stratified sediments in fluvial terraces, caves, or open-air sites, and where careful sampling avoids light exposure. OSL provides an age for the deposition of the sediment that encases the tooth. Again, it must be integrated with stratigraphic observations to ensure the tooth was not reworked. Paleomagnetism and geomagnetic reversals. Earth's magnetic field has flipped and shifted in polarity and intensity over geological time. Some sediments record the direction of the magnetic field at the time of deposition. Identifying a known reversal, for example, the Brunhes-Matuyama reversal at around 781,000 years ago, provides a powerful tie point. Paleomagnetic stratigraphy is most valuable when combined with other dating because it gives relative markers rather than absolute single-year ages. Tephrochronology, volcanic ash layers, tephra, are often compositionally distinct and can sometimes be traced to specific eruptions with known ages. A thin ash layer above or below an archaeological horizon can provide an isochronous time marker across wide regions. Tephra matching is a robust cross-check when the geochemical fingerprint is distinctive and when the eruption's age has been securely determined. Cosmogenic nuclide burial dating. For some open-air sites and surface deposits, burial dating using cosmogenic isotopes, for example, circumflex 10b and circumflex 26al, can estimate how long sediments have been shielded from cosmic rays, that is, how long ago they were buried. These methods are specialized and require careful sampling geometry, but they can extend age control where other methods are scarce. Amino acid racemization, AAR. AAR measures the slow chemical conversion of L amino acids to their mirror image D forms after death. This conversion is temperature sensitive and therefore must be calibrated against known age samples from the same thermal history. AAR can provide relative age information and is sometimes used as a supplementary check, but its sensitivity to burial temperature and diagenetic conditions makes it less reliable as a standalone absolute clock for deep time. Bringing methods together, stratigraphy and Bayesian synthesis. No single date is usually sufficient. Instead, field scientists collect multiple independent age estimates, UT on flowstones, ESR/U series on teeth, OSL on sediments, and perhaps tephra layers or paleomagnetic markers. These results are then integrated in a stratigraphic framework, which units overlie which are in primary position, and which might be reworked. Modern practice increasingly uses probabilistic frameworks, often Bayesian age modeling, to combine these data sets. In a Bayesian model, each date is represented as a probability distribution that incorporates measurement error and method-specific uncertainties. Stratigraphic relationships act as priors. For example, a layer below another must be older. The model then produces posterior age ranges for the deposits and for particular samples, which are typically presented as a best estimate with credible intervals, for example, 95% confidence ranges. Importantly, the model exposes inconsistencies. When one date strongly disagrees with others given the stratigraphy, researchers must reevaluate sampling, potential open system behavior, or reworking. Common complications and how teams manage them. Open system behavior. Uranium can migrate into bone or teeth after burial, altering youth ages unless modeled. ESR/U series methods explicitly account for such uptake histories when possible. Diagenetic alteration. Chemical changes in sediments can alter OSL signals or cause partial bleaching. Careful sampling and multiple aliquot analyses help identify these issues. Reworking and bioturbation. Burrowing animals or later human activity can relocate small finds. Microscopic wear, breakage patterns, and spatial mapping reveal such disturbance. Thermal history. Burial temperature affects amino acid racemization and the accumulation of luminescent signals. Paleotemperature reconstructions can be factored into models. Why dating matters for proteomics. A proteomic signal is most meaningful when it sits in a robust temporal framework. If a peptide pattern suggests an unexpected phylogenetic placement, the age model will either make that placement plausible or call it into question. For example, placing a specimen within a lineage that did not exist at the modeled time would prompt re-examination of stratigraphy, resampling, or reanalysis of contamination possibilities. Chronology thus serves as both anchor and test. It constrains evolutionary narratives and helps rule out post-depositional mixing or modern intrusion. Ethical note, brief. Some dating methods require removing speleothem layers or sediment blocks. These actions are destructive at a different scale than sampling a tooth, and therefore demand additional consultation with stakeholders and heritage authorities. Minimally invasive coring and careful archiving of removed materials are now standard best practices. As you let these technical points settle, hold the image of a layered profile with dates written in soft pencil next to each horizon. In the next part, we will examine a case study. Enamel proteomics from a European Pleistocene site, and what the proteins there suggested about early Homo lineages. Dim the room a fraction more, and imagine the dawn over a limestone gorge in northern Spain. Wind moves through scrub. A lead-colored river thinks about its course. In a narrow cave entrance, a thin trench lays open like a pale line in the earth. That place holds a small tooth that will speak, in a language of peptides, about a human who lived many hundreds of thousands of years ago. We begin with place and time. The specimens commonly associated with the name Homo antecessor come from a sequence of deposits in a cave system where careful excavators have worked for decades. The site's layers preserve repeated human and animal presences, and long basal deposits that reach deep into the Middle Pleistocene. For our purposes, the crucial fact is this: the tooth we follow is not merely an object on a shelf. It has a stratigraphic address, a layer placed by careful field notes, dated through flowstone, luminescence, and other anchors to the broad window of the mid to early late Pleistocene, a time when multiple branches of the human family were stirring across Eurasia. The lab receives the specimen the way labs always do: wrapped, photographed, and accompanied by the chain of custody that traces its journey from trench to bench. The enamel is assessed for preservation, cleaned of superficial contaminants, and micro-sampled with the minimal intervention required to extract peptides. The proteins targeted in enamel studies are familiar names to the attentive ear: amelogenin, enamelin, ameloblastin, structural and matrix proteins that guided the building of the crown when that tooth formed in childhood. These proteins are not abundant in a mature tooth, but fragments can be tightly locked within crystalline mineral and therefore survive where softer biomolecules have long since dissolved. What emerged when analysts read those fragments was a whisper of sequence variants, short amino acid stretches that, when compared to modern reference proteins and to inferred ancestral states, offered a pattern. In plain language, some of the peptide signatures recovered from the enamel showed affinities that made researchers re-examine where Homo antecessor might sit on the human tree. The technical claim was not that the proteome delivered a full genome or an irrefutable coup de grace to prior morphological classification. Rather, it provided molecular characters that could be compared across species, narrowing the range of plausible placements. How should we interpret such an outcome? Start with 3 quiet rules. First, the signal's quality matters. Proteomic datasets in these cases are often low in coverage. That means analysts may recover a handful to a few dozen peptides that are confidently identified and taxonomically informative. Each peptide can contain one or more amino acid positions that differ between taxa. When several peptides consistently point toward a similar placement, the inferential weight rises. But when the dataset is sparse, a single taxon-informative peptide, or peptides from regions of protein that tend to be conserved, the claim is naturally tentative. Second, reference and model assumptions shape results. Proteomic comparisons map observed peptide sequences onto a backdrop of sequences from living species and from computationally inferred ancestors. If the reference set omits a lineage with unusual sequence evolution, or if models fail to account for postmortem chemical changes accurately, inferences can be biased. Analysts therefore run sensitivity checks. They test different reference trees, they evaluate alternative placements, and they ask whether deamidation or other modifications could mimic a true sequence variant. Third, concordance with other evidence is the gold standard. Proteomic placements do best when they align with morphology, stratigraphy, and reliable dates. If enamel peptides suggest a relationship that is consistent with skeletal features and with the age of the deposit, the combined case strengthens. If the molecular signal and the morphological story diverge, that divergence is important. It flags a locus of further work, rather than an immediate rewrite. In the case of Homo antecessor, the proteomic evidence invited cautious reevaluation rather than a simple rewrite. Some analysts interpreted the enamel peptides as indicating affinities that could place the specimens near the base of certain later Eurasian lineages. Others urged restraint, noting that the data were fragmentary and that additional comparative material was needed. As with many early molecular forays into Middle Pleistocene hominins, the strongest claims were hedged. This is a piece of the puzzle, not the full picture. We should also be explicit about how labs guarded against false positives. Multiple technical strategies reduce the chance that a modern protein masquerades as ancient: strict cleanroom handling, extraction blanks processed concurrently, replication of peptide identifications across independent extracts, and evaluation of damage patterns that are consistent with long burial. Genuine ancient peptides tend to show characteristic chemical signatures, elevated deamidation levels, and other diagenetic marks that modern contaminants lack. When peptides carry these signatures and come from internal enamel rather than external surfaces, confidence is higher. Another axis of caution is chronology. The modeled age of the deposit constrains what evolutionary scenarios are plausible. A proteomic placement that implies the presence of a derived lineage too early would be immediately suspect unless the dating evidence could support such a revision. Conversely, when the chronology comfortably allows for symmetry or for ancestral branching, the proteomic signal finds a temporal home. A further layer is replication and community scrutiny. Proteomic claims that persist are those that independent teams can reproduce, or where reanalysis of spectral data by other groups supports the same interpretations. The community values openness, archiving raw spectral files, and offering transparent reporting of search parameters, because reproducibility undergirds trust. Finally, there is the human side. Sites and specimens come from landscapes with living communities and legal frameworks. When a study involves destructive sampling of an important tooth, responsible teams document permits, minimize material removed, and communicate with local heritage authorities. These steps are not mere paperwork. They are part of the ethical scaffolding that allows science to proceed with respect. As you settle that image, the cave mouth, the chipped tooth, the pale peptides aligning into a cautious pattern, hold the lesson in balance. Proteomics added a molecular thread to a complex tapestry. It did not demand a radical rewriting on its own. It offered an invitation to integrate, test, and refine hypotheses. In the next part, we will travel east to deep Asian deposits and large primate fragments, and consider what proteomics has whispered about other enigmatic forms, fragments associated in the literature with a giant ape lineage, and what their proteins might imply for biogeography and lineage survival. Dim the lab light and imagine a cavern of rainforest teeth, massive molars and jaw fragments stacked, cataloged, and waiting under careful covers. The name Gigantopithecus itself carries a kind of wonder, an echo of great size and deep-time mystery. For decades, its story has been told mostly from jaws and teeth recovered in Southeast Asian contexts, often in cave deposits or karstic sediments. These massive dental remains raise immediate questions about lineage, ecology, and the limits of molecular preservation in warm, wet places. Begin with the fossils. Gigantopithecus is known primarily from large mandibular fragments and very big molars. The morphology is bold, thick enamel, expansive chewing surfaces, and where that suggests a diet of tough, abrasive material. Because cranial and postcranial remains are scarce, investigators have long relied on dental anatomy and comparative functional morphology to infer relationships. Early comparative anatomists saw affinities with living great apes. Many suggested a close relationship to orangutans, while others left the possibility open that Gigantopithecus was a distinct, now extinct ape lineage adapted to the particular ecologies of Pleistocene Asia. The difficulty with DNA here is immediate and practical. Tropical climates are hostile to ancient DNA preservation. Heat, moisture, and microbial activity conspire to break down nucleic acids rapidly. As a result, direct genomic evidence for deep-time Asian apes has been essentially absent. That absence leaves room for proteomics to step into the gap, but it also shapes the limits of what proteomics might achieve. Proteomics asks a different chemical question. Are there enduring peptide fragments trapped within the mineral of enamel or the collagen of dentine that carry taxa-informative sequence differences? For a large molar like those attributed to Gigantopithecus, enamel is a promising target because it is the most mineralized tissue and because such teeth are commonly what survive. If enamel peptides survive, they can be read and their sequence variants compared against reference sequences from living apes and other primates. Practically, however, the tropical burial environment poses challenges. Elevated temperatures accelerate diagenetic reactions that modify amino acids and produce damaged signatures. Humid soils foster microbial colonization that can alter protein profiles, and the chemistry of karstic deposits may invite post-depositional mineral exchange. These factors reduce the likelihood of recovery and complicate interpretation when peptides are recovered. Still, enamel's mineral shelter sometimes offers a better shot than bone or dentine. If proteomic extractions from Gigantopithecus enamel yield peptides, what might they say? A few guarded possibilities emerge. First, proteins can test whether Gigantopithecus shares derived sequence traits with living orangutans. If multiple, independent enamel peptides contain amino acid residues shared uniquely with pongo, orangutans, and not with African apes or other primates, That pattern would strengthen arguments for a close relationship. Conversely, if peptide sequences place the specimens outside the pongo clade or display unexpected combinations of residues, the inference may suggest a distinct branch that split earlier or evolved along a separate trajectory. Second, proteomics can provide chronological and biogeographic context when combined with dating. If proteomic affinities suggest similarity to a lineage known elsewhere at particular times, that may inform hypotheses about dispersal routes and regional evolutionary dynamics. For example, molecular affinities consistent with orangutans might fit into scenarios of Asian ape diversification and survival in Southeast Asian refugia during glacial cycles. But the interpretive path is narrow and cautious. A few important cautions temper excitement. 1. Low coverage matters. Often only a small number of peptides are recoverable from a specimen. Each peptide may contain only one or two positions that vary across taxa. When the dataset is sparse, random errors, post-mortem chemical changes, or incomplete reference sampling can push analyses toward misleading placements. Analysts, therefore, avoid grand claims based on single peptides. They seek concordance, multiple peptides, independent damage patterns, and replication across extracts or neighboring teeth. 2. Reference bias is real. Many primate genomes are well sequenced, others are not. If the reference set lacks a lineage that once lived in Asia and whose protein sequences differed in surprising ways, the recovered peptides might best match the closest available sequence in the database rather than the true ancestral state. This is a general problem in ancient molecular studies. The inference is only as good as the comparative backdrop. 3. Diagenetic mimicry. Chemical modifications, deamidation, oxidation, or other alterations can change peptide masses and occasionally mask true residues or create misleading signals. Rigorous modeling of expected diagenetic profiles and the search for damage signatures are therefore essential to separate ancient signals from post-burial chemistry. 4. Ecology and function intersect. Dental proteins are structural. Their sequences are sometimes constrained by function. That means some positions change slowly across evolutionary time. Proteomics is therefore often better at resolving deeper, older divergences than very recent splits. Interpreting results therefore requires phylogenetic perspective. A peptide difference that seems small in absolute terms may still be significant if it occurs at a diagnostic site. Beyond phylogeny, teeth reveal ecology. Microweir patterns and isotopic signatures preserved in enamel speak of diet. A proteomic study that combines protein sequences with enamel isotope data and microwear analyses can offer a richer portrait. Lineage plus lifestyle. In Gigantopithecus, very thick enamel and certain microwear patterns have long been associated with a diet of tough, fibrous plants, perhaps including bamboo. If proteomics were to place the genus close to the orangutan lineage, that ecological data would shade interpretations of how ape lineages adapted to Asian forest mosaics. Ethical considerations arise here too. Many Gigantopithecus specimens are curated in museums and collections, with histories stretching back decades. Destructive sampling requires permission, minimal intervention, and transparent reporting. Furthermore, paleontological finds may sit on lands associated with living communities. Engagement and stewardship are part of responsible science. Finally, the role of proteomics in cases like Gigantopithecus is not to deliver final answers, but to open new windows. Even tentative peptide evidence can reframe questions, prompting renewed excavations, targeted dating programs, and comparative sequencing in living primates. The value lies in triangulation, a piece of molecular evidence guiding where to look harder, how to refine comparative samples, and which hypotheses to test next. As you close this part, picture a massive molar under a microscope, its ridges worn by chewing, its enamel-bearing microscopic time capsules. In the next part, we will step back from case studies and think about the scaffolding that makes proteomic comparisons possible across specimens, the construction and curation of proteomic reference libraries, and why those libraries are the quiet infrastructure of paleoproteomics. Dim the light and picture a long cabinet of samples, each labeled, each with its own small story. In paleoproteomics, the cabinet is both literal and digital, the physical specimens and the vast curated libraries of protein sequences and spectra that scientists use to interpret faint peptide signals. Without these references, a peptide is a shape without meaning, In this part, we will slowly open the cabinet and look at how these libraries are built, curated, and used, and why the shape of the reference set matters deeply for the stories we can tell. At its simplest, a proteomic reference library connects sequence to spectrum. For a living species whose genome has been sequenced, the predicted protein sequences can be translated into theoretical peptide masses and fragment patterns. For many species, we also have experimental spectral libraries, collections of peptide fragmentation patterns obtained from modern tissue, analyzed on the same class of instruments used in ancient work. When an ancient mass spectrum arrives, search engines compare its peaks to the reference spectra and score matches. A close match raises the probability that the peptide sequence is the same or closely related to the reference. There are several layers to this process. First, taxonomic breadth. The bigger and more representative the reference set, the better we can place an ancient peptide. If we only compare an ancient homologous peptide against human, chimpanzee, and orangutan, any match will be forced into those boxes. If the true original belonged to a lineage not represented in the set, we risk misassignment. That is why curating broad, high-quality proteomes from a diverse array of modern primates, mammals, and outgroups is essential. We want to include both close relatives and reasonable ecological neighbors so that sequence variation can be read in its proper evolutionary context. Second, experimental spectral libraries. Theoretical matching is powerful, but observed fragmentation patterns vary by instrument and protocol. Labs therefore build experimental libraries by running known reference proteins and collecting their MS-MS spectra under controlled conditions. These real spectral fingerprints improve matching accuracy. In paleoproteomics, where peptides are often chemically modified by diagenesis, libraries that include intentionally modified peptides, or at least models of common diagenetic changes, increase the chance of correct identifications. Third, ancestral sequence reconstruction. For extinct or poorly sampled lineages, researchers sometimes infer ancestral sequences computationally from phylogenetic trees built on living species. These inferred sequences produce theoretical peptides that can be matched to ancient spectra. This is a careful, model-dependent step. The inferred sequence is only as good as the tree and the substitution model used. Researchers therefore treat ancestral reconstructions as hypotheses, not as definitive references, and test sensitivity to different assumptions. Fourth, taxon-informative peptides. Not all peptide positions are equally useful for phylogenetic inference. Some sites are highly conserved across broad clades and carry no diagnostic power. Others mutate at rates that can distinguish among lineages. Reference libraries therefore highlight taxon-informative peptides, stretches where amino acid differences map clearly onto phylogenetic splits. In reporting, analysts often emphasize these peptides while being transparent about the larger background of conserved sequences. Fifth, data quality and false discovery control. Matching spectra to sequences produces scores, and analysts set thresholds to control the false discovery rate, FDR. In modern proteomics, a common FDR threshold is 1% at the peptide level. Ancient work often imposes even more conservative criteria, especially for taxa-informative calls. Laboratories also require replication, the same peptide identified independently in multiple extracts, or the presence of several taxa-informative peptides before making strong claims. Sixth, de novo sequencing and manual curation. Sometimes a peptide spectrum does not match any reference. De novo sequencing algorithms attempt to infer the amino acid sequence directly from the spectrum. These inferences are probabilistic and benefit from expert manual curation, especially when diagenetic modifications alter expected masses. Skilled analysts visually inspect spectra and consider alternative residue calls, cross-checking with phylogenetic plausibility and damage profiles. De novo results can flag missing references or novel ancient variants that merit follow-up by targeted sequencing in modern relatives. Seventh, metadata and archiving. A useful reference library is not only sequences and spectra, it is the metadata, specimen provenance, tissue type, instrument settings, extraction protocols, and the known thermal history of the sample. Archiving raw spectral data and processing parameters allows independent reanalysis and helps identify lab-specific artifacts. Open data practices, sharing spectra, and search parameters strengthen the field by enabling reproducibility and collective critique. Eighth, software and machine learning. Modern search engines incorporate models of diagenetic modification and probabilistic scoring. Emerging approaches use machine learning to predict fragmentation patterns or to flag likely ancient damage signatures. These tools help, but they also require careful training data. If the machine is fed biased references, it learns biased patterns. Continued expansion of high-quality, taxonomically diverse training sets is therefore vital. Reference bias is an important, recurring caution. When a database overrepresents certain lineages, for historical or logistical reasons, recovered peptides will tend to appear closest to those well-sampled taxa. The remedy is twofold: expand comparative sampling across biodiversity, and adopt conservative interpretive frameworks that test alternate placements rather than accepting the nearest database hit as decisive. Community standards are another cornerstone. Best practices now encourage deposition of raw mass spectrometry files in public repositories, clear reporting of search parameters and FDR thresholds, and the inclusion of extraction blanks and damage assessments in publications. These norms allow others to rescore spectra with different assumptions, to try alternative reference sets, and to test the robustness of taxonomic claims. Finally, think about living collections and ethical sourcing. Building reference libraries often requires tissue or genomic resources from modern animals and humans. Researchers must obtain samples ethically, with proper permissions, and in ways that respect conservation and human subjects' protections. Partnerships with museums, field biologists, and indigenous communities are part of responsible reference building. The diversity of the reference library should reflect ethical commitments as much as scientific ones. As you let this settle, imagine a map of the world dotted with sampling sites, museum cabinets, and sequencing centers, all contributing to a growing, collaborative infrastructure. The better the references, the clearer the whisper of an ancient peptide becomes. In the next part, we will open a myth versus evidence box, a widely repeated story about ancient lineages that protein data has helped to test, and we will carefully separate folklore from what the molecules actually say. Dim the room and settle into a steady breath. We will take a small myth, a story told often in museum captions and speculative essays, and hold it up gently to the evidence. Myths can be useful. They shape questions and focus attention, but science must test them, not accept them by habit. The myth we'll examine here is a simple, persistent one, that fossils like the European Middle Pleistocene specimens, often grouped under names such as Homo antecessor, are direct, straightforward ancestors of modern humans. The story is tidy. Find a fossil with some familiar facial traits, slot it as an ancestor, and narrate a linear progression toward Homo sapiens. It feels satisfying. It comforts the human love for ancestry that marches in neat lines. Pause and listen to that comfort. Now, put it beside what we actually know. Origin stories rooted in linearity emerged in part from early paleontology's limitations. For much of the 20th century, researchers had only morphology and fragmentary stratigraphic contexts. Morphology is powerful. Bone shape, tooth form, and cranial vault all hold evolutionary signals, but it is subject to convergent evolution and to the limited advantage of fragmentary remains. A robust nose, a cheekbone, or a dental arcade can look modern for functional reasons that do not imply direct descent. Against this backdrop, molecular evidence promised order. DNA, when available, provides detailed genealogical signals. Ancient genomes have delivered beautiful, fine-grained pictures of recent population history. They showed us admixture, migrations, and unexpected cousins. Naturally, some began to imagine a single molecular arbiter that would settle ancestry debates for older fossils as well. But two limits complicate that hope. First, DNA rarely survives beyond several 100,000 years, except in rare, cold places. For fossils 1,000,000 years old or more, DNA is usually gone. Second, morphology and partial genomes do not always agree. Where genome-scale evidence exists, it sometimes upends neat morphological placements. That mismatch fuels both revision and controversy. Enter proteomics, the quieter molecular voice. Proteins preserved in enamel and dentine can survive where DNA does not. They do not deliver entire genomes, but they can provide taxa-informative characters, amino acid positions that differ across lineages and that can be read even from fragmentary peptides. This possibility reframes the myth. Rather than forcing fossils into tidy ancestor boxes, we can test whether molecular characters support a direct ancestor scenario or instead point to branching, side lineages, or unexpected relationships. Let us walk through how myth meets evidence in practice. Take the scenario where morphology suggests a specimen is modern-like enough to be an ancestral candidate. In that case, proteomic extraction might recover enamel peptides carrying sequence variants. If those peptides consistently match the predicted ancestral state leading toward the Homo sapiens lineage, that would strengthen the ancestor hypothesis. The molecular evidence would act as an independent line of support. But if the recovered peptides instead show affinities that place the specimen outside the clade that would lead to Homo sapiens, for example, near an older Eurasian branch or aligned with other Pleistocene hominins not on the direct line, then the ancestor story weakens. Proteomics thus functions as a clarifying test. It increases or decreases the probability that a fossil is on the direct path to us. A useful way to think about this is probabilistically. Morphology assigns likelihoods, as do stratigraphy and dating. Proteomics modifies those likelihoods. Rarely does it yield certainty. Rather, it pushes the balance of evidence. When multiple lines, morphology, robust dating, and independent molecular characters converge, our confidence increases. When they diverge, we are alerted to complexity, reticulation, convergent features, misinterpretation of context, or gaps in our comparative databases. There are several illustrative patterns worth noting. First, concordance strengthens claims. If enamel peptides, morphological traits, and chronological placement all point the same way, a previously speculative ancestor claim becomes more plausible. That is the ideal conservative path. Independent lines agreeing increases trust. Second, discordance invites reinterpretation. If morphology suggests closeness to a later lineage, but proteins place the specimen elsewhere, scientists must revisit field context, re-examine stratigraphy, and assess possible contamination or diagenetic alteration. Discordance is not a defeat. It is a methodological compass directing further inquiry. Third, the absence of molecular data does not validate the myth. Silence is not confirmation. Many mid-Pleistocene fossils simply do not preserve informative proteins. For those, morphology and context remain the only available evidence, and humility should mark our inferences. Fourth, interpretive humility is essential because proteomics itself has constraints. Enamel proteins provide information from particular genes and at particular sites. The resolution is coarser than a full genome. That means proteomic data are excellent for testing broad placements and rejecting certain hypotheses, but they may lack the nuance to discriminate among very recent sister taxa or fine-scale population dynamics. Recognizing that boundary prevents overreach, let us consider a hypothetical example in plain terms. Imagine a fossil tooth from a cave layer dated reliably to 800,000 years ago. Morphology suggests a facial structure similar to later Eurasian hominins, leading to an ancestral claim. Proteomic analysis recovers several enamel peptides. Their amino acid variants align more closely with a branch that diverged earlier than the lineage leading to Homo sapiens. The conservative conclusion is not that morphology was wrong, but that the specimen likely belongs to a side branch that shared some convergent facial features. The ancestor myth, in that case, would be revised to a more branching narrative. This dynamic, myth tested by evidence, illustrates a deeper philosophy of science. Claims should be proportional to evidence. Proteomics does not romanticize a single tale. It demands that our stories accommodate multiple lines of data. When myths persist despite counter-evidence, that persistence speaks less to the robustness of the myth than to our psychological comfort with tidy narratives. Practical consequences follow. Museums and textbooks often compress complex phylogenetic uncertainty into tidy timelines. Proteomic findings encourage curators and educators to present branching, contingent histories rather than linear progressions. For listeners like you, that means embracing a more plural, nuanced human story, one in which many lines lived, mixed, and sometimes vanished, rather than a single uninterrupted march to modernity. Before we close, a quiet list of what would shift this balance decisively in a particular case. Multiple taxa informative peptides from the same specimen with consistent damage profiles. Independent replication of those peptides in different labs. concordant morphological reanalysis showing shared derived traits, and a secure chronology that places the specimen at the right moment for direct ancestry. When those elements align, ancestral claims gain real strength. In the next part, we will move from myth testing into a focused exploration of proteomics' role in shaping phylogenetic trees, how faint molecular signals are integrated into evolutionary frameworks, and why model assumptions matter. Dim the light and imagine a branching map drawn slowly in the sand, each stroke careful, each twig of the tree labeled with a question mark. Phylogenies are hypotheses drawn from many kinds of evidence. Proteomics has joined that conversation by adding molecular characters from specimens too old for DNA. In this part, we will gently trace how those faint protein signals get folded into trees, and why the results are often described with cautious language rather than trumpet fanfare. Start with the problem proteomics seeks to address. Deep-time fossils are often fragmentary. Morphology is rich, but sometimes ambiguous. Similar ecological pressures can produce similar bones or teeth in unrelated lineages. Proteomic peptides add independent characters, molecular differences encoded at the amino acid level, that, when informative, can test competing tree placements. But the path from a peptide spectrum to a tree placement is long and probabilistic. Let us walk it step by step. From peptide to character, each confidently identified peptide provides one or more amino acid residues at specific positions. Those residues can be treated like morphological characters, different states among taxa. Analysts assemble matrices in which rows are specimens, ancient and modern, and columns are character positions, peptide sites. They then use phylogenetic methods, maximum likelihood or Bayesian inference, to estimate the tree that best explains the observed distribution of states under a chosen model of sequence evolution. Model choice matters. Protein evolution models encode assumptions about how likely residues are to change and which substitutions are more probable. In deep time contexts, conservative models that allow for varying rates across sites and for diagenetic miscalls, for example deamidation, are preferable. Bayesian frameworks are particularly useful because they allow researchers to incorporate prior information, for instance stratigraphic age ranges or morphological priors, and to produce posterior probability distributions over trees rather than a single best tree. Those posterior distributions neatly express uncertainty. The tree is not a single carved fact, but a range of plausible histories weighted by their probability. Now the caveats that make cautious language necessary. Limited character sets. Unlike whole genomes, proteomic datasets often contain a small number of informative sites. A tree built on a handful of positions lacks the resolving power of one built on thousands of loci. Small datasets increase the chance that stochastic error or convergent changes at a few sites will distort inferred relationships. Researchers therefore emphasize that proteomic placements tend to resolve deeper, older splits where accumulated differences are larger, and that they are less reliable for resolving recent fine-scale divergences. Reference and taxon sampling. The tree you infer is sensitive to which modern sequences you include as references. If key lineages are absent from the reference set, an ancient peptide may be pulled toward the closest available match, even if it is not its true relative. Good practice therefore expands taxonomic sampling and tests sensitivity by rerunning analyses with different reference subsets. When placements shift dramatically with reasonable changes to the reference set, confidence is low, long branch attraction, and compositional bias. If a lineage has accumulated many substitutions or unusual biochemical biases, standard phylogenetic methods can mistakenly group it with another long-branched lineage, a statistical artifact called long branch attraction. Protein datasets are not immune to this effect. Analysts try to mitigate it with models that allow among-site rate variation, with exclusion of saturated positions, and with the inclusion of intermediate taxa to break long branches. Diagenetic miscalls and damage. Chemical alterations after burial can change masses and lead to incorrect residue assignments if not properly modeled. For example, deamidation converts neutral residues into acidic ones, changing mass and potentially producing a sequence call that looks like an evolutionary substitution. Thoughtful search parameters, damage-aware scoring, and examination of peptide-level damage profiles reduce this risk. But residual uncertainty remains. Integration with morphology and dates. The most persuasive phylogenetic inferences come from concordant signals across independence lines. A proteomic placement gains strength if it aligns with derived morphological traits and with a chronology that makes the placement plausible. Conversely, discordance prompts reevaluation. Perhaps the specimen was misdated, perhaps diagenesis altered peptides, or perhaps morphological convergence fooled earlier interpretations. In practice, integrated inference, combining morphological matrices with proteomic characters in a joint analysis or using one line as priors for the other, produces more robust, tempered conclusions. Cautious examples to illustrate the balance. Example 1. A Middle Pleistocene European tooth, hypothetical, representative. Proteomic extraction yields several enamel peptides with residues that, in broad strokes, pull the specimen nearer to later Eurasian hominins than to African lineages. Morphology is ambiguous. Some facial features are modern-like, others are primitive. The chronology places the specimen roughly 600,000 years ago. Rather than declaring direct ancestry, researchers report increased support for placement within a Eurasian radiation and stress that additional peptides, independent replication, and expanded reference sampling are needed before asserting a direct ancestral lineage to modern humans. Example 2. A large Asian molar attributed to a giant ape. A small set of peptides is recovered that match some residues in orangutan sequences, but only at conserved sites. Because the data are sparse and tropical diagenesis could confound calls, authors present the match as consistent with a possible shared ancestry or convergent conservation, not as definitive proof. They call for targeted sequencing of modern Asian primates and for attempts at replication from additional fossils. Example 3. A fragment from a poorly sampled clade. Proteomic data produce a novel residue combination not present in reference databases. Researchers cautiously consider three possibilities: an unrecognized extant-like lineage, an ancestral residue state not sampled in living species, or an analytic artifact. They treat the result as an invitation to expand reference sequencing and to search for replicated peptides in other specimens from the same stratigraphic context. Practical norms that sustain caution. Replication, independent repeats, ideally in different labs, are highly valued. Damage profiling, reporting deamidation and other diagenetic marks is standard practice. Sensitivity testing, rerunning phylogenies with alternative models and reference sets is required. Open data, sharing raw spectra and search parameters so others can reevaluate claims. Integration, presenting proteomic results alongside morphology and dating, not in isolation. A final gentle thought: phylogenetic trees are not monuments, but maps that change with each new datum. Proteomics does not so much redraw the tree in a single stroke, as it tilts local branches, nudges probabilities, and clarifies previously shadowy regions. When multiple independent lines converge, those nudges accumulate and can produce meaningful shifts in our understanding. Until then, the field's watchword is humility. Interpret probabilistically, replicate rigorously, and integrate widely. In the next part, we will turn explicitly to uncertainties, a focused examination of contamination risks, diagenetic ambiguity, model sensitivity, and the kinds of additional data that would resolve them. Dim the light and let your attention move slowly across a small bench where a handful of tubes sit like careful questions. Every confident sentence in paleoproteomics sits on a bed of uncertainty. To know the limits is as important as knowing the data themselves. In this part, we follow those limits gently and systematically, not as a list of failures, but as an honest map of the places where evidence thins and where further work is most useful. Begin with contamination, the most human of worries. Modern proteins are everywhere, on skin, in breathing, in laboratory reagents, in handling tools. A single stray peptide from a technician's glove can look alarmingly like a legitimate ancient fragment if the dataset is sparse. The practical remedy is slow ritual, strict cleanroom practice, gloves changed often, dedicated ancient biomolecule facilities, and blanks that follow the sample through every step. But ritual is not enough. The convincing sign of authenticity is pattern. Ancient peptides typically carry chemical scars of age, elevated deamidation, racemization trends, fragmentation consistent with long exposure to burial chemistry. When peptides come with those damage signatures, and when the same taxa-informative residues appear in independent extracts or in neighboring teeth from the same stratigraphic context, the probability that they are genuine rises. Conversely, peptides that lack damage, that appear in extraction blanks, or that match common lab contaminants must be treated as suspect. The field's posture is prudently Bayesian. New peptide evidence nudges our degree of belief, but it rarely overturns established inferences by itself. Next, diagenesis, the slow chemistry of the grave. Proteins do not simply vanish, they chemically transform. Deamidation converts neutral residues into acidic ones. Oxidation alters side chains, and crosslinks tie peptides into forms that confuse mass readings. These transformations create two problems. One, they can mask the original residue and produce ambiguous sequence calls. Two, they can mimic genuine evolutionary substitutions, if not modeled carefully. The laboratory response is methodological. Search algorithms explicitly permit common diagenetic modifications, damage-aware scoring is used, and analysts inspect spectra by eye when possible. Still, modeling diagenesis is an imperfect science. Thermal history, soil pH, microbial communities, and episodic wetting all alter the pace and character of chemical change, and these variables differ from site to site. Thus, a peptide profile trusted in a cold, dry cave may not translate straightforwardly to a humid karst deposit. A close cousin of diagenesis is differential preservation. Not all proteins survive equally. Enamel matrix proteins may persist while other tissues do not. Collagen may last in one setting and vanish in another. This uneven preservation imposes a kind of sampling bias. The genes we can query are a non-random subset of the genome, and their evolutionary signals are filtered through functional constraints. Some positions in a protein are highly conserved because changing them would break function. Others vary. The result is that proteomics often resolves deep splits better than fine-tuned recent divergences. Recognizing the limits of the characters at hand prevents overinterpretation. Reference bias compounds this problem. The trees we infer depend on the taxa and sequences we include in our comparative set. If databases overrepresent certain modern species and underrepresent others, for example, if wild Asian primates are poorly sampled, then an ancient peptide may be drawn to the closest available database match, rather than its true kin. The antidote is an investment in comparative diversity. More genomes, more proteomes, better sampling of living biodiversity, and careful sensitivity testing, in which analyses are repeated with alternative reference subsets to see how placements shift. Statistical and model uncertainty is another quiet place of caution. Phylogenetic inference from proteins relies on substitution models that make assumptions about rates and patterns of change. Small data sets are especially sensitive to model misspecification. A handful of taxa-informative residues can sway likelihood calculations disproportionately if the model does not account for rate heterogeneity or compositional bias. Practically, researchers explore multiple models, use Bayesian frameworks to express posterior probabilities instead of single-point trees, and report the robustness, or fragility, of results under alternative assumptions. Honesty about low posterior support or wide credibility intervals is a sign of scientific care, not weakness. Chronological uncertainty reaches in and touches all of the above. A molecular signal that suggests a derived placement is meaningful only if the chronology allows it. Reworking, post-depositional intrusion, or misdated matrices can produce apparent paradoxes. A specimen that seems too modern for its layer, for example. This is why integrated dating, multiple methods, Bayesian age modeling, and careful stratigraphic assessment is not optional. A proteomic result that conflicts with a robust, independently supported chronology must trigger reevaluation of sampling, context, and lab protocols. Then there is the problem of low coverage and statistical power. Many paleoproteomic datasets are fragmentary, a few peptides, perhaps covering short stretches of a few proteins. Each peptide offers limited characters. The probabilistic inference from such small data must be guarded. In these cases, the best scientific posture is to present results as provisional. This specimen's recovered peptides increase support for placement X relative to alternatives Y and Z, but the support is weak, and additional data are required. Communicating uncertainty transparently to peers and the public prevents premature rewriting of narratives. What kinds of additional data resolve these uncertainties? The path forward is pragmatic and plural. Replication matters. Independent extracts from the same specimen and independent analyses in different labs reduce the chance of lab-specific artifacts. Expanded sampling matters. More teeth from the same stratigraphic unit, more specimens from adjacent sites, and more modern reference proteomes fill in comparative gaps. Multi-proxy concordance matters. When proteomic signals align with morphology, secure dating, and isotopic or micro-wear indicators, the combined inference is stronger. Finally, development of improved damage models and machine learning tools trained on high-quality ancient spectra will refine residue calling and reduce diagenetic miscalls. Ethical and curatorial practices also shape credibility. Transparent reporting of permits, minimal destructive sampling, and community consultation are not peripheral. They are part of the evidentiary chain. A sample obtained and documented ethically carries additional scientific weight because it reduces the risk that provenance errors or contested curation blur interpretation. We close this part with a gentle restatement. Uncertainty is not an obstacle to progress. It is the terrain on which careful progress is made. Each ambiguity points to a precise technical test or an empirical gap. The field advances by converting those uncertainties into questions that can be answered with replication, broader sampling, better models, and open data. In the next part, we will continue examining uncertainties, focusing on phylogenetic placement sensitivity, model assumptions, and the practical experiments that would most quickly clarify disputed branches in the tree. Dim the light and picture a small cluster of trees sketched on vellum, each branch labeled with a probability rather than a certainty. Phylogenetic placement from sparse protein data is a statistical conversation with limits. In this part, we keep the voice steady and practical. What makes placement sensitive, how researchers test those sensitivities, and what concrete experiments narrow the uncertainty. Begin with the mathematical heart of the matter, models and priors. Phylogenetic inference depends on an explicit model of how amino acid residues change over time. Different models make different assumptions about substitution rates, exchangeabilities among residues, and the distribution of rates across sites. For tiny data sets, a handful of peptides covering a few protein sites, these model choices exert outsized influence. A model that assumes homogeneous rates across sites will treat each observed difference differently than one that allows some sites to evolve faster. Bayesian methods add priors that can include stratigraphic age or morphological expectations. These priors can stabilize inference, but, if misspecified, can also bias outcomes. Sensitivity testing, running analyses under alternative models and priors, is therefore essential. When an inferred placement collapses or flips under reasonable model changes, that fragility must be reported. Next, consider missing data and limited character sampling. Proteomic datasets commonly sample only a subset of the proteome and often only conserved structural proteins. Those proteins are functionally constrained. Many positions change slowly and provide little discriminatory power for recent splits. Conversely, conserved positions can still be valuable for very deep divergences. The result is uneven resolving power. Practically, researchers treat each amino acid position as a character with its own information content and often weight conclusions toward placements supported by multiple, independent characters rather than single-site calls. Long branch attraction and taxon sampling follow closely. When a lineage has accrued many substitutions, or when key intermediate taxa are missing from the reference set, methods can mistakenly group fast-evolving or poorly sampled lineages together. Good taxon sampling breaks long branches and reduces this artifact. In practice, investigators test robustness by adding or removing reference taxa, by including outgroups, and by experimenting with reduced data sets to see whether the target specimen's placement is stable. Alignment and homology are another foundational uncertainty. Proteins must be aligned correctly so that homologous residues are compared. Short damaged peptides increase ambiguity. A misaligned peptide can introduce false differences. Analysts inspect alignments manually when possible, use multiple alignment algorithms, and report sites that were ambiguous or excluded. Conservative reporting often limits claims to placements supported by clearly homologous, well-aligned sites. Diagenetic modification intersects with model sensitivity. Chemical changes after burial, deamidation, oxidation, cross-linking can alter observed masses and confound residue calls. Analysts compensate by including likely modifications in search parameters and by modeling expected damage profiles. Yet two related problems remain. First, damage can produce apparent substitutions that look evolutionary but are not. Second, different burial environments produce different damage regimes. So, a damage model trained on cave samples may not generalize to tropical karst. Sensitivity analyses that vary the allowed set of modifications and comparisons across specimens with similar thermal histories help reveal whether placement is robust or driven by diagenetic artifacts. Statistical support and its interpretation deserve careful attention. Bootstrap values, posterior probabilities, and likelihood ratios all quantify support, but small datasets can produce deceptively high numbers when data are shallowly informative. A high posterior probability, based on a few correlated sites, is not as persuasive as modest support that emerges across many independent characters. The community's best practice is to report measures of support transparently and to contextualize them. Explain how many characters underpin a clade and whether support changes under alternative models. Model misspecification and compositional bias also matter. If the observed residues in an ancient sample reflect compositional shifts, perhaps due to diagenesis favoring certain modified residues, standard models that assume stationary base frequencies may mislead. Testing models that allow non-stationary composition or applying recoding strategies, reducing the alphabet to broader residue classes, can reduce bias. These techniques are especially useful when small numbers of sites dominate the signal. What are the practical experiments and analyses that most rapidly clarify ambiguous placements? Several concrete steps stand out. Expand comparative sampling. Sequence proteomes and genomes from underrepresented modern taxa that are plausible relatives. Filling gaps in the reference library reduces misassignment risk and breaks long branches. Replicate in independent labs. Independent extractions and blind reanalyses reduce the chance that lab-specific contamination or processing idiosyncrasies drive a result. Targeted enrichment. When peptides of interest are suspected but coverage is low, targeted capture of specific proteins or peptides, analogous to targeted DNA capture, concentrates signal and increases the number of informative sites recovered from a specimen. Simulated degradation experiments. Bury modern teeth or enamel under controlled conditions, varying temperature, pH, and microbial exposure, and sequence the resulting peptides over time. These experiments generate empirical damage models that improve sequence calling and reveal which diagenetic pathways produce misleading residue patterns. Joint morphology molecular analyses. Combine proteomic characters with morphological matrices in a single, integrated Bayesian framework. Such integrative models allow morphological data to inform branch lengths and priors while letting molecular characters pull placement where they support it. Concordance across data types strengthens inference. Sensitivity grids. Systematically explore the analytic parameter space. Vary substitution models, rate heterogeneity parameters, damage allowances, and taxon subsets. Reporting the range of placements across this grid clarifies which conclusions are robust and which are parameter sensitive. Cross-validation and leave one out tests. Remove single peptides or sites and rerun analyses to see whether any single character disproportionately drives placement. If a removal materially changes topology, the result is fragile. Improve damage-aware scoring. Invest in software that models diagenetic chemistry empirically, using training sets derived from controlled degradation and high-quality ancient spectra. Better models reduce miscalls and increase confidence. Communication and reporting norms are the final human layer. Clear statements of the data's character. How many peptides, which proteins, how many informative sites, damage profiles, independent replication, and sensitivity tests allow readers to judge the evidence. Avoiding categorical language and preferring probabilistic phrasing, this evidence increases support for X relative to alternatives Y and Z, prevents overclaiming, and fosters cumulative science. We end this part with a practical, modest invitation. When you read a proteomic placement, look for the scaffolding. How many characters support the claim? Were alternative models tested? Was there independent replication? These are the signs of careful work. In the next part, we will shift from models and uncertainty to ethics and stewardship, the principles that guide destructive sampling, reporting, and collaboration with descendant communities and curating institutions. Dim the light and breathe slowly. Before we take any further molecular step, we must sit with an ethical truth. These are not only specimens of scientific curiosity, they are often the remains of once living people or creatures, and they come from places held by living communities. Ethical practice shapes every methodological choice, from whether to drill a sliver of enamel to how results are reported and shared. In this part, we hold ethics not as an add-on, but as a structural element of responsible science. Begin with permission and provenance. Responsible research starts long before a drill touches a tooth. It begins with clear documentation of where a specimen came from, who excavated it, and under what permits it was removed. In many jurisdictions, cultural heritage laws regulate excavation and export. Researchers must follow those legal frameworks. Equally important are the communities who live near sites, indigenous peoples, local stakeholders, and national heritage authorities. Good practice includes prior consultation, transparent explanation of the research goals and possible destructive impacts, and written agreements that describe what will be removed, how much will be taken, and how remnants will be curated. Minimal destructive sampling is a core principle. Proteomics often requires only a tiny chip of enamel, the size of a grain of rice. Modern protocols emphasize microsampling techniques, careful documentation of the removed portion, and preservation of the remaining specimen for future study. Sampling plans typically specify the minimal volume needed to achieve scientific objectives and describe how the remaining materials will be stored. Museums and repositories increasingly require researchers to justify destructive sampling with clear scientific necessity and to provide plans for returning unused fragments to the collection. Consent and community partnership matter. In regions where human remains have spiritual or ancestral significance, consultation is not mere courtesy. It is ethical imperative. Researchers should engage with communities early, explain methods and goals in accessible language, and heed cultural sensitivities. Sometimes communities will permit sampling only under certain conditions. For example, involvement of local researchers, co-authorship, or shared access to data. In other cases, communities may request that sampling not occur. Respecting those decisions is part of scientific humility. Some knowledge is not ethically worth obtaining at the cost of violating living people's values. Transparency and reciprocity are practical corollaries. When communities permit research, reciprocal benefits should be tangible. These can include capacity building, training local students in sampling or analysis, sharing results in accessible formats and languages, contributing to local museums or education programs, or returning curated items after analysis. Benefits should be negotiated, documented, and sustained beyond a single paper or press release. Good partnership is not extractive. It is collaborative and ongoing. Legal frameworks and repatriation add further layers. Some collections contain materials whose removal or export was conducted under colonial-era arrangements now regarded as unjust. Increasingly, institutions and researchers are engaging in repatriation dialogues and provenance research to correct past harms. When sampling is proposed on such collections, curators, legal teams, and communities must work together to ensure that research does not perpetuate historical inequities. Sometimes the ethical path leads to restraining research until rightful ownership and stewardship are clarified. Data stewardship and open science carry ethical weight as well. Sharing raw spectral files, analytical parameters, and curated results enables reproducibility and communal verification. Yet openness must be balanced with sensitivities. Genetic or proteomic data from human remains may touch on privacy, identity, and cultural protocols. Where appropriate, data sharing agreements can limit misuse and ensure that access respects community preferences. Many projects now include controlled access archives or tiered sharing plans that honor both scientific transparency and cultural concerns. Reporting and interpretation are ethical acts. Language matters. Scientists should avoid sensational headlines that reduce complex, probabilistic findings to certainties. Instead, results should be presented with clear statements of uncertainty, methodological limits, and the cultural contexts of the specimens. When findings have implications for living communities' histories or identities, authors should discuss interpretive limits and avoid making claims that could be misread as definitive assertions about ancestry or entitlement without robust, multi-line evidence and appropriate consultation. Museums and curatorial practices are part of the stewardship chain. Museums increasingly require that sampling proposals include plans for post-analysis curation, how the remaining specimen will be stored, what documentation will accompany it, and how future researchers can request access. Curators may also impose restrictions on destructive analysis to preserve the specimen's integrity. Collaboration with curators ensures that the scientific value of sampling is balanced against the specimen's long-term cultural and research value. Ethical review and oversight mechanisms help translate principles into practice. Institutional review boards, heritage authority approvals, and museum committees provide checks and balances. These bodies evaluate whether the scientific aims justify destructive sampling, whether community consultation occurred, and whether data stewardship plans are adequate. Researchers should anticipate these reviews and prepare thorough documentation, sampling rationale, methods, storage plans, and community engagement records. Benefit sharing and authorship deserve practical attention. When local collaborators contribute materially to fieldwork, curation, or interpretation, their intellectual contributions should be recognized. This can include authorship on papers, acknowledgments, or explicit co-ownership of datasets. Benefit sharing extends to capacity building, funding student exchanges, supporting local labs, or contributing to heritage conservation projects are meaningful, practical ways to reciprocate. Finally, consider the precautionary principle. If the scientific benefit is marginal and the destructive impact non-negligible, the ethical choice may be to refrain. Science thrives on curiosity, but it must also recognize limits. Preserving a specimen intact for future technologies that could extract more information less destructively is often the more respectful option. As we close this ethical chapter, imagine a laboratory bench not just as a place of instruments and tubes, but as a table at which multiple voices, curators, local communities, legal authorities, and scientists are represented. Ethical practice is the listening that precedes, accompanies, and follows the technical work. In the next part, we will return to methods with a deeper technical dive, peptide sequencing nuances, crosslinks, deamidation patterns, and practical lab experiments that sharpen signal from noise. Dim the lights a fraction more, and let your breath settle into a slow, even rhythm. In this part, we move deeper into the lab's small, technical chamber. We will listen closely to how a mass spectrometer reads the muffled voices of ancient peptides, how those voices are changed by time, and what practical steps scientists take to separate signal from the din. Begin with the extracted peptide mixture, the small powder or micro drillings freed from inner enamel and brought into solution. What arrives at the instrument is rarely a single clean peptide. It is a complex soup, short endogenous fragments, broken bits of larger proteins, chemical adducts formed during burial, and trace modern contaminants. The laboratory's task is to make that soup intelligible. Proteolysis and the question of enzyme specificity. In modern proteomics, it is common to digest proteins with a protease like trypsin. Trypsin cleaves predictably after lysine and arginine, producing peptides of a typical length that mass spectrometers handle well. For ancient samples, digestion is a double-edged sword. On one hand, controlled digestion can generate peptides of predictable boundaries that improve database matching. On the other, much of the endogenous material in ancient enamel is already heavily fragmented. Additional digestion can obscure naturally occurring termini that themselves carry information about age and preservation. Some workflows therefore apply minimal or no enzymatic digestion to preserve endogenous cleavage patterns. Others use gentle digestion to increase identifiability. The choice is deliberate and justified in method sections. Liquid chromatography and tandem mass spectrometry, LC-MS-MS. Peptides are separated by liquid chromatography to reduce complexity, then ionized and injected into the mass spectrometer. The instrument measures the mass-to-charge ratios, NMZ, of intact peptide ions, and then isolates particular ions to fragment them. Tandem mass spectrometry records the masses of these fragments, producing spectra that are diagnostic of the peptide sequence. Fragment ions are commonly labeled as B ions and Y ions, fragments that include the N-terminal or C-terminal portion of the peptide respectively, and their pattern across the spectrum is the laboratory's primary evidence for residue order. Database search, scoring, and false discovery. Interpreting spectra happens in two main ways. In database searching, the observed spectra are compared against theoretical spectra derived from reference protein sequences. Search engines score peptide spectrum matches, and statistical procedures estimate the false discovery rate, FDR. Because ancient peptides are often chemically modified, search parameters must allow for expected mass shifts, deamidation, oxidation, etc., and for non-tryptic termini when appropriate. Conservative thresholds and the use of decoy databases help control false positives. De novo sequencing and manual curation. When spectra do not match any reference, de novo sequencing algorithms infer the most probable amino acid sequence directly from fragment masses. De novo calls are inherently probabilistic and benefit from expert inspection. In paleoproteomics, skilled analysts visually inspect spectra, check for consistent ion ladders, and evaluate whether a proposed sequence is phylogenetically plausible given references. De novo results can indicate novel ancient variants or gaps in the reference library. They can also reveal artifacts that a blind search might mistakenly accept. Damage patterns and diagenetic modifications. Time changes amino acids in predictable ways. Two common chemical transformations are especially diagnostic. Deamidation, asparagine, N, and glutamine, Q, residues slowly convert to aspartic acid, D, and glutamic acid, E, respectively. This change increases peptide mass by about +0, 984 Da, and accumulates with burial time and elevated temperature. Elevated deamidation rates at internal residues are strong indicators that peptides are ancient rather than modern. Oxidation, methionine, and other residues oxidize, +15, 994 Da for methionine oxidation. Oxidation patterns can reflect both slow burial chemistry and handling or laboratory exposure. Other modifications, crosslinks, hydrolytic cleavage, advanced glycation end products, AGEs, produce more complex mass shifts and can obscure simple sequence calling. Crosslinks in particular create peptides that are covalently bonded to one another, producing higher mass species and complicated fragmentation patterns that standard search engines may not handle well. Specialized cross-linking aware workflows and targeted analyses are required to interpret these spectra. Racemization and chirality. Amino acid racemization, the conversion of L to D forms, is an important chemical clock, but it does not change peptide mass. Detecting racemization requires separate chemical assays that measure DL ratios. Such assays complement mass spectrometric sequencing by providing an independent preservation metric. High DL ratios support the antiquity of recovered peptides, but racemization rates depend heavily on thermal history. Proteotypic and taxon-informative peptides. Not every peptide is equally useful for taxonomy. Proteotypic peptides are those that are reliably detected and map uniquely to a protein or taxon. Taxon-informative peptides include residues that differ across lineages and thus carry phylogenetic signal. Analysts prioritize such peptides when assembling character matrices for phylogenetics. A single unique peptide change at a diagnostic site can be powerful, but only if authenticated by damage profiles and replication. Targeted mass spectrometry, PRM, SRM, MRM, and isotopic standards. When shotgun, untargeted proteomics yields tantalizing but sparse signals, targeted MS methods can increase sensitivity. Parallel reaction monitoring, PRM, or selected multiple reaction monitoring, SRM, MRM, focuses the instrument on specific precursor fragment ion pairs, improving detection limits for known peptide sequences. Heavy isotope-labeled synthetic peptides can be spiked into samples as internal standards, allowing absolute quantification and confirming retention times and fragmentation patterns. For ancient work, synthetic peptides encoding both the endogenous sequence and expected modified variants help validate identifications. Cross-validation, blanks, and replication. Authentication requires multiple lines of laboratory control. Extraction blanks, procedural blanks, and environmental controls sail through the workflow. If a peptide appears in blanks, it cannot be trusted. Independent replication, extracting a second aliquot from the same specimen or analyzing a nearby tooth from the same layer, is a powerful authentication. When different labs, using different instruments and protocols, converge on the same peptide identifications with similar damage patterns, confidence rises substantially. Spectral networks and cluster analysis. Emerging tools construct spectral networks that group related spectra, revealing families of peptides that share common fragment patterns or modifications. These networks can detect modification series, cross-linked products, or degradation ladders, and can flag spectra that warrant manual inspection. Clustering also helps distinguish recurrent modern contaminants from true ancient signals by pattern matching across datasets. Statistical reporting and probabilistic thinking. Finally, the field frames results probabilistically. Analysts report the number of peptide spectrum matches, PSMs, the number of distinct peptides, which proteins they map to, measures of modification frequency, for example, percent deamidation, and whether peptides are unique to a taxon. Phylogenetic claims are expressed as increased support or shifted probabilities rather than categorical declarations. This cautious language reflects the inherent limitations of short modified peptide datasets. As you let this technical detail settle, imagine the spectrum on a screen, a series of peaks, a ladder of fragments, and a small team of quiet analysts turning those peaks into a narrative, slowly, conservatively, and with layered checks. In the next part, we will look outward again. What proteins reveal about diet, disease, and mobility, and how molecular traces in teeth can illuminate the lived experience of past individuals. Dim the light and imagine a single tooth held up to the spare beam of a desk lamp. It is small, but inside its creases and mineral lattices are traces of a life. What was eaten, what microbes lived in the mouth, perhaps a hint of childhood disease, and even a whisper of how that body moved across landscape. Proteins do not narrate entire biographies, but they carry fragments of the lived experience. Short sentences that, when stitched together with other evidence, form a quieter, more intimate portrait. Begin with dental calculus, the hardened plaque that forms on tooth surfaces. Calculus is a sticky, mineralized archive. It traps food particles, microbes, host proteins, and environmental debris. Once mineralized, it resists decay and can persist for millennia. Proteomic analysis of dental calculus is one of the most direct ways proteins record diet and health. Dietary proteins. Proteomics has detected food proteins preserved in dental calculus that directly indicate what individuals ate. Examples include milk proteins, beta-lactoglobulin and caseins, which are robust markers of dairying and milk consumption when identified and authenticated with damage profiles. Finding milk proteins in calculus provides direct evidence that a person consumed dairy and, depending on age at enamel formation and the tooth sampled, can sometimes suggest childhood weaning practices. Plant proteins, cereal storage proteins, legume albumins, or specific seed proteins can also survive in calculus and point to plant consumption that might not leave macroscopic traces. Proteins are particularly valuable because they can distinguish between food sources that produce similar isotopic signatures. For instance, certain cereals and tubers may produce overlapping carbon isotopes, but their proteins are taxonomically distinct. Oral microbiome and disease. The mouth is an ecosystem. Calculus traps bacterial proteins and fragments of microbial cells, providing a proteomic snapshot of the oral microbiome at the time of burial. Shifts in microbiome composition reflect diet, hygiene, and disease. Proteins of periodontal pathogens, or markers of dysbiosis, can indicate chronic oral inflammation or infection. Some studies have recovered proteins from pathogens associated with systemic disease, though pathogen protein detection is more challenging than bacterial DNA due to low abundance and degradation. Still, when pathogen peptides are recovered with appropriate damage signatures and controls, they offer direct evidence of infection that complements genomic or isotopic signals. Host immune proteins. Embedded in calculus and sometimes preserved in dentine are fragments of host proteins involved in immunity and inflammation. Defensins, salivary enzymes, and immunoglobulin fragments. These proteins can indicate oral immune responses to infection or stress and may hint at chronic conditions. For example, elevated presence of certain inflammatory proteins across multiple teeth could suggest prolonged periodontal disease. Interpreting immune proteins requires careful baselines. Modern oral proteomes vary with age, diet, and other factors. So, comparative modern reference sets help contextualize ancient findings. Weaning and childhood life history. Teeth grow in incremental layers. Different parts of the crown form at known childhood ages. When dietary proteins or isotopic signals are mapped to specific enamel formation intervals, for example, first molar enamel forming roughly from about birth to 3 years, researchers can infer childhood diets. The presence of milk proteins associated with enamel or calculus from early forming teeth suggests breastfeeding or milk consumption during specific developmental windows. When combined with enamel incremental lines that timestamp growth, proteomic traces can map dietary shifts onto individual life histories, weaning ages, early introduction of complementary foods, or episodes of nutritional stress, sexing and biological identity. Proteomic approaches have developed methods for sex determination in situations where DNA is absent or degraded. Enamel contains amelogenin, a protein encoded by genes on both X and Y chromosomes, AMLX and AMELI. The presence of AMELI-specific peptides can indicate male individuals. Proteomic sexing has proven useful, especially for juvenile remains where morphological sexing is unreliable. However, the method depends on recovering the Y-specific peptides reliably and on excluding contamination. The absence of a Y peptide does not prove femaleness if preservation is poor. The approach is powerful when combined with other lines of evidence, and as with all proteomic inferences, requires replication and clear reporting of peptide coverage, mobility, and life course movement. Proteins are not primary tracers of mobility the way strontium or oxygen isotopes are, but they contribute indirectly. Enamel proteins tell us when a part of a tooth formed, when those time-stamped proteins carry dietary markers, for example, milk proteins or specific plant proteins, that differ from later formed regions. We can infer shifts in diet that sometimes align with movement, migration, seasonal mobility, or cultural change. More directly, calculus proteins capture oral microbiomes shaped by local diets and environmental exposures. Abrupt shifts in microbiome composition between teeth that formed at different ages may reflect changes in residence or diet tied to movement. Integrating proteomic signals with isotopic mobility markers produces the clearest picture. Isotopes track landscape, proteins hint at foodways and microbiomes, and together they map movement and lifestyle. Pathogen detective work. Detecting pathogen proteins, fragments from bacteria, viruses, or parasites, is rare but potentially informative. Pathogen proteins are typically low abundance and degrade rapidly, but in mineral-protected matrices or calculus they can sometimes be recovered. When authenticated, pathogen peptides provide direct evidence of infection and can sometimes point to disease type. For deep-time contexts, pathogen protein evidence remains sparse, and claims must be treated cautiously. Complementary evidence, lesions on bone, ancient DNA, epidemiological context, strengthens pathogen inferences. Functional and metabolic clues. Some preserved proteins report on host metabolism. Enzymes involved in saliva production, digestive function, or oral biochemical processing can indicate dietary regimes or physiological adaptations. For example, expansion of salivary amylase gene copies in some populations relates to starch consumption. Proteomic signals of amylase in dental calculus can corroborate such dietary niches. Interpreting metabolic proteins requires careful cross-cultural modern baselines and attention to post-mortem modification. Limitations and interpretive caution. Proteomic inferences about diet and disease are powerful but selective. Not all food types preserve equally. Oily fish or certain meats may leave less proteinary trace than milk or cereal grains. Low abundance, diagenetic alteration, and contamination all complicate interpretation. Moreover, the presence of a food protein in calculus records consumption, not necessarily regular diet. It could reflect a single meal, ritual food, or occasional specialty. Triangulation with isotopes, microfossil residues, like starch grains or phytoliths, dental microwear, and archaeological context gives the most robust picture. Ethical note, brief. Interpreting diet and disease touches on living descendants' histories and identities. Researchers should communicate findings with humility, avoid over-interpreting single data points, and engage communities in discussing implications for health narratives, ancestry claims, or cultural heritage. We close this part with a human image, a child's tooth bearing milk proteins, an adult molar with evidence of a plant-rich diet, and a calculus layer telling of microbes and meals long digested. Proteins translate ephemeral acts, feeding, illness, migration, into strands of evidence that, when combined carefully, let us glimpse the textures of past lives. In the next part, we will bring proteomics, ancient DNA, and morphology into a joined conversation, a multi-proxy synthesis that shows how each line of evidence strengthens or qualifies the others. Dim the light and settle into a slow breath. We have listened to enamel, traced peptides, worried about diagenesis, and held ethical care in our hands. Now let us bring the different threads into a single loom. Proteomics is most powerful, not as a solo instrument, but as part of an ensemble. Ancient DNA, morphology, isotopes, stratigraphy, and archaeological context each sing a part. In this part, we follow how those voices are combined, how conflicts are resolved, and what a careful, integrative conclusion looks like. Begin with the principle of complementarity. Each method samples a different dimension of the past. Ancient DNA, when available, provides broad genomic sweeps, high-resolution signals of ancestry, admixture, and population dynamics. Morphology records the phenotype shaped by genetics, development, and environment. Proteomics captures conserved molecular characters from tissues that often survive where DNA does not, and it can also carry direct life history signals like sex markers or dietary proteins. Isotopes and microfossils add environmental and mobility context. Together, these lines reduce the chance that any single artifact, contamination, or misinterpretation drives a large conclusion. A practical workflow in integrative analysis often follows three phases: independent assessment, joint modeling, and iterative refinement. Phase 1. Independent assessment. Each dataset is analyzed on its own terms with protocols suited to its strengths. Morphologists score traits blind to molecular results. Proteomic analysts report peptides, damage profiles, replication, and taxon-informative residues. Geneticists present genome-wide affinity patterns or fragmentary aDNA reads with contamination estimates. Dating teams provide Bayesian age models integrating UTU, ESR, OSL, and stratigraphy. Each group states uncertainties explicitly, posterior probabilities, confidence intervals, and the number of supporting characters are documented. This independent rigor ensures that subsequent integration is not simply a blending of biased inputs. Phase 2. Joint modeling and cross-validation. Integration proceeds through statistical frameworks that can weight evidence and express joint uncertainty. Bayesian hierarchical models are particularly useful. They allow molecular characters, morphological characters, and chronological priors to enter a single analysis where the posterior distribution reflects all inputs. In practice, researchers might use a morphological matrix to anchor topology and run molecular characters as additional data, or vice versa. Sensitivity analyses test how conclusions shift when specific data sets are downweighted or excluded. If a proteomic signal strongly shifts topology only when a particular assumption is made, that fragility is made explicit. Cross-validation is an important humility test. If proteomics suggests placement A, while morphology supports placement B, researchers ask which inference is more sensitive to assumed priors, to taxon sampling, or to damage models. They may then seek additional data targeted to resolve the discrepancy: more teeth from the same layer, targeted peptide enrichment, further anatomical study, or more refined dating. The goal is not to force an agreement, but to map precisely where evidence aligns and where it does not. Phase 3. Iterative refinement and open reporting. Science advances by iteration. A tentative integrated placement generates hypotheses for additional data collection. Teams publish raw spectra, morphological matrices, and dating models so others can reanalyze with alternative assumptions. When independent groups replicate results, finding the same peptides, the same morphological synapomorphies, and concordant dates, confidence increases. When conflicts remain, publications explicitly present alternative trees or rank hypotheses by posterior support, rather than presenting a single definitive diagram. A few concrete patterns emerge from integrative work. Concordant convergence. The clearest cases are those where morphology, proteomics, and chronology point the same way. Here, the cumulative probability can justify stronger language. A specimen is likely to belong to clade X with Y% posterior probability, supported by Z number of peptide sites and N morphological synapomorphies. These are the moments when paleontology and molecular paleobiology jointly rewrite local branches of the tree. Molecular rescue. Proteomics sometimes rescues otherwise ambiguous fossils by adding independent characters that tip the balance. A tooth with equivocal morphology, but with several taxa-informative peptides, can move from uncertain to probable placement, particularly in regions where DNA is lacking. Discordant tails. Occasionally DNA, when available, and morphology disagree with proteomics. These cases demand careful forensic work. Was the specimen reworked? Is there lab contamination? Are the molecular references inadequate? Or might morphology reflect convergent adaptation? The forensic process, redating, replication, expanded references, often resolves the dispute or reframes the question in a more nuanced way. Practical examples of synthesis, conceptual, not citing specific papers. A mid-Pleistocene tooth with enamel peptides that slightly favor a Eurasian radiation is integrated with a cranial fragment, showing mixed features. Combined Bayesian models that include both data types produce a posterior that favors a branching near the base of Eurasian hominins, but with broad, credible intervals, prompting targeted searches for additional specimens in the same horizon. A tropical molar yields dietary proteins and calculus, plant and starch signatures, that align with microwear and isotope signals pointing to a plant-rich diet. Proteomics confirms dietary hypotheses that morphology had suggested but not proven. Guidelines for weighting evidence in integrated inference. Demand replication for molecular claims before they sway morphology-driven interpretations. Prioritize concordance with secure chronology. A molecular signal that conflicts with tight dating requires extraordinary scrutiny. Use explicit probabilistic language. Report how much each line of evidence shifts posterior probabilities. Be transparent about reference biases. Show how placements change with different comparative sets. Communication to broader audiences requires care. Integrated conclusions should avoid sensational certainty. Instead, narrate the balance of evidence. Current data increase support for X, but alternative Y remains plausible given Z uncertainty. This language invites the public into the provisional, cumulative nature of science rather than offering settled truths. We close with a soft invitation. When you read an integrated study next, look for the scaffolding. Independent assessments, joint models, sensitivity checks, and clear statements of what more would resolve remaining doubts. Integration is not the end of uncertainty. It is the method by which we narrow and clarify it. In the next part, we will look ahead. Future technologies and approaches that may deepen proteomics' reach, from targeted enrichment to improved damage models and hybrid molecular morphometrics. Dim the light and let your attention settle on a clean bench where instruments hum like distant seas. If paleoproteomics has taught us one thing, it is this. Small technical gains can open large interpretive doors. In this part, we move gently into the near future, not as futurists making grand promises, but as careful observers imagining the methods, experiments, and infrastructures most likely to sharpen the faint voices in ancient teeth. We will keep our feet rooted in the practical, what is plausible, what is already emerging, and what would most effectively reduce uncertainty. Begin with sensitivity and targeted recovery. One of the bottlenecks of ancient proteomics is simple: low abundance. The peptides that matter are often present in minute quantities and obscured by background material. Borrowing a page from ancient DNA work, targeted enrichment approaches are already being adapted for proteins. These methods use designed capture reagents, synthetic peptide baits, antibody pull-downs, or molecularly imprinted polymers, to fish out peptides of interest from a complex mixture. Targeted mass spectrometric methods such as parallel reaction monitoring, PRM, or multiple reaction monitoring, MRM, SRM, then interrogate those candidates with far greater sensitivity and quantitative rigor. In practice, a lab might design capture reagents for a set of taxon-informative enamel peptides plus control peptides that report damage. Pulling those targets from a tiny enamel extract can multiply the effective information recovered from a single chip. Top-down and middle-down proteomics are another promising direction. Instead of digesting proteins into short tryptic peptides, top-down methods analyze intact proteins or large fragments. This preserves labile post-translational modifications and endogenous cleavage patterns that carry information about age and diagenesis. Middle-down approaches, analyzing larger peptide fragments, strike a balance between identifiability and preserving contextual signals. Applied cautiously to well-preserved enamel fragments, these approaches could increase the number of informative residues recovered and better differentiate diagenetic modifications from true sequence variants. Instrumental advances will matter too. Mass spectrometers continue to increase in resolution and dynamic range. Ion mobility separations add an orthogonal dimension, separating peptides not just by mass but by shape and charge, helping disentangle complex salt and mineral-rich extracts. Data-independent acquisition, DIA strategies record more comprehensive fragment information for each run, making it easier to reinterrogate datasets later as spectral libraries and search strategies improve. Together, these enhancements reduce the chance that a low-abundance, taxa-informative peptide is simply lost in noise. Machine learning and improved damage models are quietly transformative. Deep learning models trained on large sets of modern and experimentally degraded peptides can predict likely fragmentation patterns, anticipate common diagenetic mass shifts, and improve de novo sequencing of spectra that lack exact matches and references. Importantly, training these models requires high-quality datasets that include controlled degradation experiments. See below. With better trained models, analysts can more reliably separate genuine ancient residues from chemical artifacts and propose candidate sequences that merit targeted follow-up. Controlled degradation experiments are an infrastructural priority. Burying modern teeth in enamel under known variable conditions, different temperatures, moisture regimes, microbial communities, and sediment chemistries, and then periodically analyzing recovered peptides produces the empirical data needed to build robust, environment-specific damage models. These experiments convert speculation about diagenesis into quantifiable parameters. They also improve our ability to predict which burial contexts are most likely to preserve informative proteins, aiding field sampling strategy. Non-destructive and minimally destructive sampling will grow in importance. Museums and communities rightly prefer that specimens be preserved. Innovations in microsampling, ultra-fine robotic micro-drilling, laser-assisted microablation that removes sub-milligram quantities, and glue lift methods that recover surface molecules without visible loss, reduce the impact of analysis. Coupled with enhanced sensitivity and targeted capture, these methods mean more data from less material. Parallel developments in micro-CT guided sampling, where three-dimensional imaging pinpoints the best micro-locations for extraction, reduce unnecessary removal, and maximize the yield of endogenous molecules. Cross-omics integration will become more seamless. Proteomics will pair with fragmentary aDNA, epigenetic marks, isotopes, and morphology in joint statistical models that treat each data type as a probabilistic contributor. Software pipelines that natively analyze proteomic spectra alongside short ADN reads and morphological matrices will reduce ad hoc synthesis and increase transparency. In practice, this means joint Bayesian models that can weight weak proteomic signals against stronger morphological priors, updating posterior probabilities as new targeted peptides or new dates are added. Single-molecule protein sequencing is an aspirational technology to watch. Early-stage methods aim to read individual protein molecules without digestion by measuring electrical or optical changes as amino acids pass a nanoscale pore or through other detection schemes. If and when robust single-molecule protein sequencing becomes practical for degraded, cross-linked ancient material, it could obviate some of the ambiguity introduced by complicated fragmentation patterns. That said, This technology remains experimental and will require major adaptation for mineral-bound, chemically modified peptides. Field portable analyses and faster feedback loops will shift sampling strategies. Portable mass spectrometers and lab-in-a-container units are improving. While current portable instruments do not match high-resolution lab systems, they can triage samples in the field, detect gross preservation, identify promising contexts, and prioritize specimens for full lab analysis. This triage reduces wasteful destructive sampling and helps centers without immediate lab access decide which tiny samples to export or analyze locally. Reference expansion and stewardship remain foundational. No method will substitute for a globally representative, ethically sourced library of modern proteomes and experimental spectra. Building that library means partnerships with museums, field biologists, and communities to sample modern biodiversity responsibly and to deposit spectral data in open, curated repositories with appropriate access controls. It also means archiving raw spectra and metadata from ancient runs to allow future reanalysis with improved models. Ethical and governance technologies will evolve alongside methods. Data access frameworks that incorporate community preferences, controlled access repositories for sensitive human-derived proteomic data, and standardized consent protocols for destructive sampling are emerging practices. Future work will likely formalize benefit-sharing mechanisms tied to datasets and create clearer expectations for capacity building in countries and communities that steward the sites. Finally, practical experiments that would most accelerate progress include coordinated global burial experiments across climatic gradients, interlab ring trials where identical enamel fragments are distributed blind to measure reproducibility, targeted capture pilot projects focused on a small panel of taxa-informative enamel peptides, and investments in open spectral libraries with well-annotated damage profiles. These are pragmatic steps that convert methodological promise into reproducible practice. As you let these possibilities settle, picture a field technician in a rain-softened trench deciding which tiny tooth to sample, guided by an on-site triage instrument, an ethical agreement filed in the team's satchel, and a protocol that will remove only a microgram of enamel. Picture later, in the lab, targeted probes pulling out a handful of peptides whose spectra are read with high confidence by an instrument that watches fragments in 3 dimensions. Picture the data flowing into a joint model that gently nudges a branch on the tree and prompts a decade's worth of new field questions. This is not a single dramatic revolution. It is a steady sharpening of tools and norms. In the next part, we will step back to list 3 to 5 specific unsolved puzzles, the crisp questions that, if answered, would change how we read proteins in teeth and rewrite key parts of the deep human story. Dim the light and breathe slowly. Here we gather a handful of the clearest, most consequential puzzles that sit at the intersection of teeth, proteins, and the deep past. Each question is practical and specific. It names a gap, describes what would resolve it, and explains why an answer matters. Treat this as a map of high-leverage uncertainties, the places where careful experiments, ethical sampling, and modest technological gains would change how we read ancient proteins. Puzzle 1. How reliably can we distinguish ancient diagenetic change from true evolutionary substitutions? The problem. Chemical modifications after burial, deamidation, oxidation, cross-linking, can alter peptide masses and sometimes masquerade as amino acid changes. If a modified residue is misread as an evolutionary substitution, phylogenetic placement is vulnerable to error. What would resolve it? Systematic, controlled degradation experiments. Bury modern teeth and enamel under varied, well-documented conditions, temperature, moisture, pH, microbial community, and sample them over time. Generate high-quality spectral libraries that pair known original sequences with their progressive damaged forms. Train models on these empirical data so software can recognize which mass shifts likely reflect diagenesis versus real sequence variation. Why it matters. Better damage models shrink a major source of false positives and increase confidence in taxon-informative calls. This in turn lets small peptide datasets carry more honest weight in phylogenetic inference. Puzzle 2. What is the minimal set of taxa-informative enamel peptides needed for robust placement at different timescales? The problem. Proteomic datasets are often sparse. We do not yet know how many independent, well-authenticated peptide sites are required to resolve a deep split hundreds of thousands to millions of years, versus a shallow one, tens of thousands of years. Without these benchmarks, interpretations oscillate between cautious and overconfident. What would resolve it? Simulation studies combined with empirical downsampling. Take dense proteomic matrices from well-sampled modern clades, remove data to mimic ancient coverage, and measure how placement accuracy degrades. Pair these simulations with controlled enrichment experiments, targeted capture, to see how many real sites can be recovered from small samples. Publish guidelines. X authenticated peptides with Y damage profile and Z replication level produce a given posterior support threshold. Why it matters. Clear benchmarks guide field decisions, which specimens to sample, and publication thresholds, what counts as evidence robust enough to revise taxonomic placement. Puzzle 3. Can targeted enrichment panels be standardized and validated across labs for ancient enamel peptides? The problem. Targeted recovery improves sensitivity, but ad hoc panels and lab-specific pipelines create reproducibility problems. A panel that works in one facility might underperform elsewhere. Without standardization, community trust grows slowly. What would resolve it? Community ring trials. Design a small, prioritized panel of taxa-informative enamel peptides and damage control peptides. Distribute blinded, identical aliquots of degraded experimental material and of carefully curated ancient extracts to multiple labs, and compare recovery rates, false positives, and damage metrics. Produce an open versioned capture panel specification and validated protocols. Why it matters. Standardized capture panels would raise baseline sensitivity, make results comparable between groups, and accelerate confident replication of borderline findings. Puzzle 4. How deep can proteomics push reliably in tropical and subtropical contexts? The problem. Warm, wet climates are hostile to biomolecules. Yet many important fossils from human evolution come from such regions. We need to know whether enamel proteomics can be routinely informative in those climates, or whether only exceptional preservational windows will yield data. What would resolve it? A coordinated sampling program across climatic gradients that compares identical sampling protocols. Teeth and associated sediments from tropical karsts, temperate caves, dry open sites, and permafrost contexts. Combine proteomic recovery statistics with local burial parameters, thermal histories, and sediment chemistry to build an empirical preservation map. Pair this with on-site triage tools to identify promising specimens before destructive sampling. Why it matters. Knowing the preservation envelope prevents wasted destructive sampling and prioritizes sites where protein recovery is realistic. It also focuses technology development on the hardest but most rewarding contexts. Puzzle 5. How do we balance minimal destructive sampling with future-proofing specimens for technologies we cannot yet imagine? The problem. Destructive sampling is finite. Every micro-drill chip removes material that future methods might exploit more gently. The urge to answer pressing questions must be weighed against the possibility of better, less invasive methods tomorrow. What would resolve it? Agreed community standards and registry of sampled specimens, mandatory minimal sampling justifications, pre-registered protocols, archived residual fragments, and a deferred sampling option where curators restrict destructive testing until certain technology milestones are met. Coupled with investment in non- or minimally destructive methods, laser microablation, glue lifts, micro-CT guided micro-drilling, this approach protects future opportunities. Why it matters. Stewardship preserves both scientific and cultural value. It ensures that decisions to sample are made transparently and proportionally, and that future generations inherit the option to ask new questions. Cross-cutting experiments that short-circuit multiple puzzles. A few practical projects would accelerate resolution across these questions. Global burial matrix experiments, multiple sites, climates, producing matched spectral libraries. Interlab ring trials for both shotgun and targeted capture approaches. Targeted capture pilots on curated museum teeth with replicated extractions and open archiving of raw spectra. Standards for minimal sampling, and a public registry of sampled specimens and leftover fragments. A gentle ethical caveat: each experimental program must be governed by transparent consent, stakeholder consultation, and careful curatorial planning. Technical progress cannot outrun ethical practice. The two must advance hand in hand. We close this part with a modest invitation. Imagine a 10-year program that pairs controlled degradation studies, coordinated field triage, and open ring trials. The combined results would reduce ambiguity, create community standards, and make proteomic signals far more decisive. In the next part, we will pull these threads into a gentle synthesis. What we can reasonably claim now, what remains speculative, and how to live with uncertainty while remaining curious and careful. Dim the light and let your breath lengthen. We have walked from the trench at dawn to the hum of the mass spectrometer, and we have held questions about contamination, chronology, and the ethics of sampling. Now we pause to weave the threads into a careful tapestry. This part will not tie every loose end. It will instead gather the most reliable strands, mark the frayed ones, and describe, clearly and softly, what kinds of new data would strengthen or overturn current inferences. What we can say with modest confidence, teeth are small time capsules. Enamel and, to a lesser extent, dentine and dental calculus preserve molecular fragments under favorable conditions. In many burial contexts, especially cold or mineral-stable sites, enamel proteins can survive far beyond the temporal reach of recoverable DNA. When authentic peptides are recovered, they provide independent molecular characters that complement morphology and dating. Proteomics extends the molecular window. Where aDNA is absent or too fragmented, proteomic sequences from enamel give us molecular evidence for phylogenetic placement. This extension is not unlimited. Proteomics often offers coarser resolution than genomes, but it reliably contributes to deep time questions where DNA is silent. Authenticity depends on pattern, not on a single peak. The community's best practice is conservative. Multiple peptides, documented damage patterns, clean blanks, independent replication, and concordance with context are the pillars of credible claims. Single peptide reports without replication remain provisional. Integration is the path to trust. The strongest inferences arise when proteomic signals align with robust chronology and morphological traits. Where those lines converge, probability shifts meaningfully. Where they diverge, the divergence is itself informative. It tells us either that our models, references, or sampling are incomplete, or that evolution and taphonomy have produced a complex story. Proteomics is especially valuable in understudied regions. Tropical and subtropical contexts often destroy DNA, but sometimes preserve enamel proteins. This makes proteomics a crucial technique for parts of Asia, Africa, and elsewhere, where gaps in genomic data otherwise leave major parts of the human story dark. Where uncertainty remains, and why. Damage versus substitution ambiguity. Diagenetic changes can mimic evolutionary substitutions when not modeled correctly. We need better empirical damage models tied to specific burial regimes to reduce false positives. Until then, sequence calls at marginal sites must be treated cautiously. Sparse data and statistical fragility. Many datasets contain only a handful of informative residues. Small character matrices are sensitive to model choice, taxon sampling, and single-site effects. This statistical fragility means that many proteomic placements are best described as increasing support for a hypothesis rather than proving it. Reference bias. Our ability to place ancient peptides depends on how well modern proteomes represent relevant biodiversity. Undersampling of certain primate lineages or geographic populations can skew assignments. Building ethically sourced, broad comparative libraries is essential. Preservation envelope unknowns. We still lack precise empirical maps of where proteins reliably survive across climatic and sedimentary gradients. Knowing that envelope would prevent unethical sampling in hopeless contexts and direct resources to genuinely promising specimens. Ethical complexity. Not all scientifically answerable questions are ethically permissible to pursue, especially when sampling affects descendant communities or cultural patrimony. Ethical constraints rightly shape what we may and may not test. What would shift our conclusions most decisively? Replicated peptide sets from the same specimen analyzed in independent labs, showing consistent damage profiles. Multiple taxa-informative peptides recovered from a single individual, with robust chronological anchors that make the placement temporally plausible. concordant results from proteomics, morphology, and secure Bayesian age models. Expanded reference proteomes that fill current taxonomic gaps and remove nearest neighbor misassignments. Standardized, validated, targeted capture panels that reliably increase peptide recovery across labs. Practical guidance for researchers and stewards. For field teams, prioritize meticulous context recording, sample only when the stratigraphic address is secure, and obtain community permissions early. Use on-site triage when possible to avoid needless destructive sampling in poorly promising contexts. For laboratories, maintain strict cleanroom protocols, archive raw spectra and metadata, run extraction blanks, and encourage independent replication. Use damage-aware search parameters and report the full set of evidence: peptide counts, damage metrics, replication status, in publications. For curators and communities, insist on minimal sampling justifications, clear benefit-sharing arrangements, and protocols for archiving leftover material. Collaborate in advance on research questions, not after results are drafted. For readers and communicators, prefer probabilistic language. When a paper reports that proteomic data increase support for a placement, treat that as an invitation to evaluate the evidence scaffolding, rather than as a final verdict. A pragmatic, hopeful map forward, build coordinated experimental programs that pair controlled degradation experiments with field-based preservation surveys. These generate the empirical models needed to interpret diagenetic signals. Invest in ring trials and shared capture panels so that sensitivity and specificity are community validated across instruments and protocols. Expand ethically sourced modern reference proteomes, prioritizing underrepresented taxa and populations. Prioritize targeted enrichment methods and minimally destructive sampling innovations to get more from less. Foster integrative projects that combine morphology, proteomics, aDNA where available, isotopes, and rigorous dating in single Bayesian frameworks. A brief ethical reflection: science is generous when it shares benefits and modest when it admits limits. Proteomics offers new access to stories previously hidden by time, but it also imposes choices: when to sample, what to publish, how to involve custodians. Good practice requires that the pursuit of knowledge never crowd out respect for the living and the future. A final human sentence before we move to close: molecules in enamel are tiny, patient witnesses. They cannot speak alone. They acquire meaning only in conversation with rocks, bones, dates, and people. We listen, we triangulate, we hold our conclusions lightly. In the final part, we will gather a gentle closing, a synthesis for listeners who prefer the slow, calm wrap-up, and leave you with practical takeaways, a short ethical checklist, and the soft sign-off that guides this channel. Dim the light one last time and draw a slow breath. We began at a trench at dawn and moved through microscopes, mass spectrometers, and ethical conversations. We have listened to small molecules trapped in enamel and let them tug, gently and cautiously, at long-held stories about human lineages. Before we close, I will gather practical takeaways, an ethical checklist you can carry in your mind, and a quiet reflection on what this science, careful, incremental, and collaborative, asks of us. A short synthesis, softly stated: proteomic analysis of ancient teeth is not a magic lamp that reveals definitive ancestors. It is a careful sensor that reads fragments of molecular memory and translates them into probabilistic evidence. Enamel and dental tissues preserve peptides longer than many other biomolecules under the right conditions. Those peptides can add useful characters to phylogenetic questions, especially when in a is absent. But the signal is partial, sensitive to context, and best interpreted alongside morphology, robust dating, and archaeological information. When proteomics is strongest, several threads converge: secure stratigraphy and chronology, multiple, independently authenticated peptides with clear damage profiles, replication across extracts or laboratories, and concordance with morphology or other proxies. When one of these threads is weak, ambiguous context, single peptides, lack of replication, the most honest position is cautious. This evidence increases support for hypothesis A, but alternative B remains plausible. Practical takeaways for different readers. For curious listeners, Appreciate nuance. Single molecular discoveries are exciting, but science advances by accumulation. Look for transparency. Good studies report raw spectral data, damage metrics, replication, and dating models. Enjoy the slow story. The field refines hypotheses over years. Headlines compress what is often a gradual refinement. For field archaeologists and curators, record key text meticulously. Photogrammetry, precise stratigraphic logs, and sealed sampling protocols are essential. Prioritize minimally destructive sampling only when the stratigraphic and research case is strong. Expect to negotiate. Permits, stakeholder agreements, and clear benefit-sharing plans are part of modern practice. For laboratory scientists, keep strict cleanroom protocols and run blanks at every stage. Archive raw spectra and metadata. Use damage-aware search parameters and report deamidation, oxidation, and other modification frequencies. Seek independent replication and participate in ring trials to build communal trust. For communities and heritage managers, ask for clear explanations of the research aims, sampling plans, and how results will be shared. Expect reciprocity, training, co-authorship, where appropriate, and accessible summaries of results in local languages. Preserve agency. Saying no to destructive work is a valid and important choice. An ethical checklist, short and practical. Provenance documented. Make sure field records, photos, and chain of custody are complete. Minimal sampling justified, confirm sample volume, alternative non-destructive options considered, and why the science needs that material. Community consultation, evidence of meaningful engagement, and clear agreements on benefits and data sharing. Permits and legal compliance, local heritage laws and museum policies respected and documented. Data stewardship, plans for archiving raw spectra, metadata, and residual fragments are in place. Publication transparency, protocols, blanks, damage profiles, and sensitivity analyses will be reported. What good outcomes look like. Replicated, cautious publications that present probabilities, not certainties. Shared spectral libraries and open protocols that reduce duplication of effort and increase reproducibility. Ethical partnerships that build local capacity and return benefits to source communities. A gradual, clarified narrative about deep human lineages that values complexity over neat progressions. A calm word about interpretation and responsibility. It is tempting to want tidy stories, direct lines from fossil to modern human. The scientific practice we've traced resists simplicity when evidence does. Proteomics helps us move toward a more plural, nuanced history. Many branches, occasional contact between them, migrations, local extinctions, and small populations with unexpected genetic or proteomic signatures. That complexity honors the messy, real processes that made us. For storytellers and teachers, frame findings probabilistically. Avoid turning preliminary molecular results into definitive ancestry claims. Encourage audiences to value the method, how replicability, open data, and multi-proxy synthesis lead to more reliable knowledge over time. A modest call to future practice. If you are part of this work, and as a field worker, lab scientist, curator, or community partner, consider these modest priorities. Build broader and ethically sourced reference libraries. Develop and participate in ring trials. Invest effort in controlled degradation experiments to sharpen damage models. And commit to transparent data sharing with appropriate cultural safeguards. These steps convert promising methods into reliable, communal tools. A brief human reflection. The small molecules trapped in teeth connect us across deep time, not because they prove tidy narratives, but because they invite humble listening. Each peptide is a fragment of a life, a child's enamel that recorded milk, a molar microware that hints at a foraged meal, a damaged peptide that nudges our understanding of a branching tree. The reverence we bring to people, to places, and to the slow labor of evidence-making is part of the science. Parting practicalities for listeners. If this episode helped you think more clearly about how molecular evidence works, please subscribe or share. Small acts help sustain careful, slow content. If you fall asleep while listening, that is perfectly fine. The transcript and timestamps will be in the description to return to later. If you are working with sensitive specimens, the ethical checklist above is a concrete starting point for discussion with colleagues and communities. A final, quiet sign-off. Thank you for spending this long, gentle hour with me, for following small things into large questions, and for choosing curiosity tempered by care. Keep two simple habits. Listen patiently to evidence, and hold conclusions lightly. The past is rich and complicated. Our job is to honor its complexity with careful methods and respect for those who steward its traces. Stay curious, stay calm,